Check, sound check. Check, check one, check. Come in, come in. Check, check one, sound check. अब check है. थोड़ा सा नीचे कर लो उसको. Check, check one. Check, check one. A very good morning to all of you. Thank you for joining us 15th India Probiotic Symposium. Check, check one, sound check. Check. A very good morning to all of you, and I welcome all of you to 15th India Probiotic Symposium. We will be beginning very soon. Till then, make yourself comfortable and thank you for your patience.
Check, 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 check. Hello, hello. Check. Hello, hello.
For the ones who are not present in the hall but can listen to me, please join us here. We are about to begin.
Check. Hello.
I request all of you to settle down so that we can begin with today's first session. And once again, I welcome all of you to 15th India Probiotic Symposium. This is the day 2 we have gathered here for, and we're going to begin with the first session of the day. Good morning, everyone. Our esteemed chairpersons, speakers, delegates, senior international colleagues, and our young investigators for today. Now we will start our first session of the day on gut microbiota, probiotics, and interaction with host metabolism. May I invite Professor Bindu De, former Secretary, TDB, and retired advisor, DBT, Government of India, Director, Research and Development Cell, RDC, Swami Rama Himalayan University, Dehradun, Uttarakhand, India, and Professor Pankaj Malhotra, Professor and Head Department of Clinical Hematology and Medical Oncology, Postgraduate Institute of Medical Education and Research, Chandigarh, India, to chair the session. Let's have a huge round of applause for our chairpersons. Thank you.
Good morning, friends. Welcome to the first session of the second day. We had very exciting talks and lot of leads on basic science front and also on the clinical trials. And then in the evening we had wonderful Bhangra dance. So you all are feeling energetic to share your thoughts for today. We have got 3 speakers for this session. The first speaker is Dr. Javed. The second speaker is Dr. Anil Kumar. And the third speaker is Dr. Tarini Shankar Ghosh. Dr. Javed, you are the first speaker. You'll get about half an hour. And I would request all the speakers to stick to the time because many of the dignitaries would leave, for their flights in between. About Dr. Agrewal, he is primarily, he is a PhD from SN Medical College, Agra University. He was former scientist and professor, and he's currently former chief scientist and professor in IIT Ropar, if I believe. Earlier, he was in IMTech, Chandigarh. His specialization is on immunology of infectious diseases and gut microbiome. He has got about 135 publications. He is the Shanti Swarup Bhatnagar Awardee. National Bioscience Award has been conferred to her. He is having Fellowship of Medical Research Council UK, Bill and Melinda Gates Foundation Award for High Quality Research, then UP Science Award for Lifetime Achievement, etc. He's a fellow of INSA, IISc Bangalore, and Nasi Allahabad. Dr. Agrawal, please.
Thank you, Dr. Bindu De. And I'm grateful to the organizers, especially Professor N.K. Ganguly, Dr. Nija, Dr. Neenam Taneja. And today I'll be talking about the unveiling the impact of gut-mediated gut microbiota, the spices of the loss of dendritic cell tolerance. So currently I'm working at Indian Institute of Technology, Ropar. And we did this, most of the work was done at my previous institute, Institute of Microbial Technology. So first of all, I would like to thank my team, and the work today I'm going to present is mostly done by Hilal. And also it's done by Junaid. So we have been listening a lot about gut microbiota since yesterday, that it can control many things, including it can control different type of diseases associated with whatever organ you wish to see over here. And of course, recently it's been also shown that it can modulate the immune system. And the vaccine efficacy can be modulated by gut microbiota as well, and as well resistance to infections. So recently there were two very interesting papers, and I've been allotted 30 minutes, but this clock is showing 15 minutes. So recently there were in 2020, there were two very good papers, and these papers, they showed that the eosinophils, they regulate the inflammatory response which is induced by adipose tissues. And ultimately, it can have impact on immunological fitness in old age. And another interesting thing, interesting study was done that when eosinophils from young, they were transferred into the aging, and it was observed that the aging was reversed. So everybody feels like they should have, they should remain young, even though they become old. So how this aging can be delayed or aging can be reversed so they can start looking again young. So in this context, we observe, since we observed that gut microbiota can impact eosinophils, so we thought that one of the important cell of immune system is dendritic cells. So it is these cells which can only cell that can activate naive T cells into effector cells, and they get ultimately differentiated into effector T cells and memory T cells. So these dendritic cells, they are quite diverse, since they can activate the immune system, they can activate naive T cells, as well as they have tolerogenic capacity as well, so that they maintain peripheral tolerance. The cells which have escaped thymic education, and these autoreactive T cells, teeny tiny population which escape thymic deletion, they remain inside our body, and these are the dendritic cells which ultimately are responsible for keeping the cells of the body, especially like T cells and B cells, into tolerogenic state. So to keep this in mind, we thought that what may be the impact of aging on the gut microbiota? Does it induces the dysbiosis of gut microbiota? And if it does induce, then what is the impact on dendritic cells? So in that context, we started characterizing dendritic cells from young animals as well as aged animals. And then we thought, if the young animals' gut is disrupted, dysbiosis, how do these young animals' dendritic cells, they behave, whether they have capacity to activate T cells, or they can induce tolerance or activation of T cells. Can their interaction leads to NRG, unresponsive in T cells, and then we also did analysis of gut microbiota and how does it changes with age, the microbiota stratification, and ultimately whether if there is any disappearance of a particular gut microbe. And if you transfer this gut microbe into the old animals, how does they behave? So for this, what we did initially, we took animals, and from literature, we identified that people who they use, young animals, usually they are 2 months old, and the animals which are old, they are 22 to 24 months old. So same way we selected such animals, and these animals, young animals, we treated with antibiotics so that we induce dysbiosis. So initially what we did, we took bone marrow cells from these animals, and then we developed them into dendritic cells with the help of IL-4 and GM-CSF. And then we analyzed these dendritic cells. At the same time, we also analyzed the gut microbiota of these animals, and we observed that there was a substantial decline in both aerobic and anaerobic population of gut microbes in old as well as the young, which the gut was disrupted through treatment through antibiotics. So it was quite amazing to see that the aerobic, both anaerobic and aerobic bacteria, they decreased significantly. But interesting part was that when we looked into the cell number of the bone marrow cell number, we observed that there was no difference upon dysbiosis or old animals. However, these bone marrow cells, when we use them to transform into dendritic cells, so we observed there was a substantial decrease in the capacity of these bone marrow cells to get differentiated into dendritic cells, both in old as well as dysbiotic animals. So next, we thought that these dendritic cells, whether they have lost their phenotypic capacity or the functional capacity, or is there any change in the function as well. So then we monitored the important molecules, that is CD886, MSC class 2 molecules. Since dendritic cells, as I told you, they are important cells in activation of naive T cells. And to activate naive T cells, they should express optimum level of MSC molecule as well as costimulatory molecules. So interestingly, what we observed that these animals, which the old animals, they were expressing high level. There was upregulation in the expression of CD8, T86, and MSC class 2 molecules, indicating they acquire the hyperactivation activity. So next, we also looked into the expression of CD80 molecules, and we also observed that there was increase in CD40 as well. So CD40, CD80, 86, they are considered to be costimulatory molecules, and these molecules are important for the optimum activation of T cells. So TIM3 is one of the molecules which is responsible for immunosuppression. So surprisingly, what we observed that these cells, dendritic cells, they showed downregulation in the expression of TIM3, and this we observed both through flow cytometry and as well RT-PCR. Now the important thing was whether these dendritic cells have capacity to phagocytose antigen, whether they phagocytose antigen, they can they uptake antigen, and then whether they can process and present this antigen to the T cells. So whereas in young animals, we observed through flow cytometry that they could uptake substantially the antigen, and whereas the dysbiotic animals, they showed downregulation in the uptake of the antigen. And this we showed through flow cytometry as well as we showed through confocal microscopy. So you see here that in young animals, there was sufficient uptake of dextrin FITC, whereas in dysbiotic or old animals, they could not uptake much of the antigens, indicating that the dendritic cells from old animals, they also lose their capacity for antigen uptake. So next was whether they have a capacity to activate T cells. So to check the functionality of dendritic cells, we took both syngenic CD4 T cells as well as allogenic T cells. And these T cells, they were anti-CD3, CD28 activated. And then we cultured them with dendritic cells so that we provide equal physiological conditions to young as well as old T cells. So in that case, we observed that young animals, they optimally activated T cells, whereas the old and dysbiotic animals, they showed hyperactivation of these T cells. So these T cells, these dendritic cells, they could even activate the naive T cells as hyperactivation of naive T cell, and also they could break down tolerance. So the important part was that what exactly is responsible in these dendritic cells so that they are hyperactivating these T cells and breaking down tolerance of tolerized T cells. And we observed that the secretory molecules, especially IL-6, IL-1 beta, TNF-alpha, IL-12, interferon gamma, these are all secretory molecules, pro-inflammatory cytokines, they were highly upregulated as compared to young animals when they were compared with old animals or young dysbiotic animals. So the young animals, if you treat them continuously with antibiotics, their gut start behaving like old animals. And these bacteria, some of the bacteria, since they disappear, ultimately leads to generation of more of hyperactive dendritic cells rather than and decreasing the frequency of tolerogenic dendritic cells. So it was quite interesting and intriguing to note that the anti-inflammatory cytokines like IL-10, IL-2, TGF-beta, IL-4, they were highly down-regulated. So these cells, the dendritic cells, they started producing less amount of tolerance or of immunosuppressive cytokines, indicating the majority of these dendritic cells, they are of activation phenotype rather than tolerogenic dendritic cells. So besides these, using ELISA, we also confirmed our result through RT-PCR, and we could reproduce these results with RT-PCR as well. So all pro-inflammatory cytokines, they were increasing, whereas anti-inflammatory cytokines, they were decreasing. So further, we wanted to see whether these genes which are responsible in controlling or suppressing the secretion of activation molecules, and they are responsible of suppressing, or they are highly upregulated in tolerogenic dendritic cells. What is the status of those molecules? So then we start looking into IRF4, IRF8, PDL1, BTLF4, and ALDH2. Interesting part was that all these molecules, we found that they were highly downregulated in old and young dysbiotic animals compared to the young one. So these molecules are already established to play a suppressive role in dendritic cells. So this indicates that these dendritic cells, they are of high activation profile, and they shift towards more of activation profile rather than tolerogenic dendritic cells. So then we thought to look into the phenotype of T cells, especially whether it can, what is the status of regulatory T cells, since regulatory T cells are quite important in suppressing the immune system. So again, we observed that the old animals, they showed very low percentage of regulatory T cells as evidenced by the expression of FOXP3 and CD25, as well as there was high expression or high percentage of regulatory T cell in young as compared to the old. So we also, now we started looking into the mechanism. What exactly is the mechanism which gene is ultimately controlling the hyperactivation of these dendritic cells in old animals? So one of the key molecule is responsible for homeostatic signaling is NF-kappa B. So NF-kappa B, it drives the expansion of the activated cells, and these tolerogenic dendritic cells, they have a lot less of NF-kappa B. So next is that we started looking on this activation marker, NF-kappa B. So we looked into its phosphorylation of P65. So we observed that in young animals, there was very less expression of NF-kappa B, whereas there was a high expression of NF-kappa B in both young and dysbiosis. And so we also observe this through confocal microscopy by staining. It's one of the subunit P65. And if you see here that this subunit was highly expressed in dysbiotic and old animals, but not in young animals. So we also looked into these other immunosuppressive markers where they are considered to play a crucial role in case of dendritic cells like IDO and iNOS, nitric oxide, arginase. And then in this case, we observed that at the gene level, there was a downregulation in express of iNOS, which is considered to be important in providing tolerogenic capacity to dendritic cells. And then we also observed in downregulation in nitric NO and then arginase as well as IDO. So these molecules, they play a very crucial role in regulating the tolerogenic function of dendritic cells. So this, besides through RT-PCR, we also showed it through Western blot just to substantiate our data. So similar results we observed with the Western blot as well. So now, the important thing what we wanted to see is that what exactly is going on in the gut? How is the gut modulated? So we did, looked into through NSG, we looked into relative abundance of the phylum. And we observed there was a downregulation in firmicutes, and whereas there was increase in the level of bacteroids. So these bacteroids, they are considered to be important for the activation of, for activating the function of dendritic cells. So in this case, we observed that in old animals, there was high level of bacteroids, whereas less level of firmicutes. And then we looked into abundance of genus, and it was Lactobacillus, which ultimately we observed. And then we looked into the species level, and it was found to be relative abundance of Lactobacillus plantarum. So if you see here, in this case, as compared to young, there was a substantial decrease in the level of Lactobacillus. So this Lactobacillus, ultimately, we started looking into these. We took this Lactobacillus and it was identified to be Lactobacillus plantarum. So this Lactobacillus plantarum we used to gavage these animals, and then we looked into whether this gavaging can revert back the functional activity of dendritic cells. So we gave this Lactobacillus plantarum into the animals which were old, and then we compared the relative abundance in their gut of Lactobacillus plantarum. So we monitored for 3 different time points, and we observed that the animals which were not given Lactobacillus plantarum, the old animal didn't show any change in the frequency, whereas when Lactobacillus plantarum was given to these animals, there was substantial increase in the frequency of Lactobacillus plantarum in the gut of these animals. So the next is since we observed earlier that with age there is the breakdown of epithelial barrier in the intestine. So when these old animals, we provided Lactobacillus plantarum, and then through Dexton Fitzy, we observed whether it goes into the blood. So we measured this Lactobacillus, we measured Dexton Fitzy in the blood, and we could not see much in the blood. So indicating that the permeabilization or epithelial barrier, it becomes intact upon given Lactobacillus plantarum. So they start behaving like gut of young animals. So next is, then again, we started repeating same experiments as we observed in case of old animals. We gave Lactobacillus plantarum, and when we observe the expression of CD86, we observed CD8086, and then we observe that the expression of this 86 or 80, it was optimally expressed compared to young animals. So there wasn't heightened expression of this molecule, and same way we observe the expression of CD40. You see this CD40 was also not highly expressed, it was optimally expressed as seen in young animals. And same way we observe that this TIM3, which was down-regulated, it started coming up-regulated in case of old animals. So we next wanted to see the phagocytic activity of these dendritic cells. So they retained phagocytic activity upon giving Lactobacillus plantarum. And this was shown through flow cytometry as well as through confocal microscopy. So if you see here that upon feeding Lactobacillus plantarum, the old animals, they showed equally good phagocytic activity like of young animals. So old shows substantial decrease in phagocytic activity. So indicating that upon transfusion of Lactobacillus plantarum, the animals, they start acquiring the young phenotype in their gut. So same way we observed that the hyperactivation molecule of pro-inflammatory cytokines, they were down-regulated. All of these cytokines, whereas anti-inflammatory cytokines, IL-10, TGF-beta, IL-2, IL-4, they increased as compared to the old animals. So there was an increase in these cytokines. So indicating that these dendritic cells, which they started acquiring more of tolerogenic phenotype. And this was further substantiated the result through RT-PCR, the gene level, and we observed that all these pro-inflammatory cytokines, they start decreasing, whereas anti-inflammatory cytokines, they started increasing. So we also looked into the genes which are directly responsible in case of dendritic cells for inducing tolerance. So like RF4 is increase, RF8 increase, PD1 started PD-L1, PD-L2, and BTLA4 and ALDH2, all of them, these molecules, they start increasing. So these dendritic cells start acquiring a normal phenotype, a phenotype which is responsible not to break down tolerance, not to induce autoimmunity, rather than inducing optimal activation of T cells. And same way we observed other tolerogenic molecules like nitric oxide, inos, and these molecules, they are directly responsible for the activity of dendritic cells. IDO is considered to be the best molecule responsible for induction of tolerogenic phenotype in dendritic cells. So it was increased, and this was shown through RT-PCR as well as through Western blot. And you see over here, whereas the inos and IDO, they were highly down-regulated in case of old, upon Lactobacillus plantarum, that it was upregulated. So besides IDO, this arginase was also upregulated. So replenishment of the gut microbes with the Lactobacillus plantarum, it also revived the metabolic shift in these animals, as we observe the expansion through the population of NBD positive bone marrow derived cells. So this was highly increased in NBDG uptake, and this could also be observed through confocal microscopy. So then it was important to see that what is the function, what is the activity or the impact on NF-kappa B, NF-kappa beta, since it was modulating, it was the main molecule responsible for the hyperactivation of dendritic cells. And upon Lactobacillus plantarum, we observed that there was substantial decrease in the expression of the NF-kappa B, plus its other molecules like REL1A, and also subunit of NF-kappa BP365 we observe through flow cytometry as well as through Western blot. And you can see here that there was substantial decrease, and this was further reproduced by confocal microscopy. And here we could observe that there was substantial decrease upon Lactobacillus plantarum given to old animals compared to the old animals. So you can see visually that these old animals' dendritic cells are expressing high level of NF-kappa B compared to old animals were given Lactobacillus plantarum. So now the important part is upon the functionality of these T cells. So whereas these T cells were responsible for hyperactivation, and when we use same system like previous one, we cultured these dendritic cells with syngeneic CD4 T cells and allogenic CD4 T cells. And then we observed that like old cells, they were inducing hyperactivation of syngeneic as well as allogenic cells. So upon Lactobacillus plantarum, we observed that there was a downregulation in the proliferation of CD4 T cells. And most of these cells which we observed, these were FOXP3 positive. So you can see here, these were gated on CD4 positive, CD25 and FOXP3 cells, syngeneic cells. So as compared to old, there was a statistically significant increase in the level of FOXP3 cells and as well as in allogenic cells as well. So then we wanted to see whether these dendritic cells, since previously they showed downregulation in the migratory markers, especially CCR7 and CCR5, these two molecules are CCR9. These two molecules, chemokine receptors, they are considered to be very crucial in helping migration of dendritic cells. Since these dendritic cells, they had lost their capacity of migration since they were downregulated in old animals. So when Lactobacillus plantarum was given, we observed that there was remarkably there was increase in the expression of these molecules. So both CCR7 and CCR9, they were highly upregulated. So indicating that these animals, they show these young animals, they start expressing high level of activity in dendritic cells. Same way we observed in the expression of FOXP3, that is in old animals which were given Lactobacillus plantarum, we observed that they acquired activity. And then we did transfer experiments from old animals treated with Lactobacillus plantarum. We transferred these regulatory T cell into young animals, and we observed that these animals, they start behaving like normal animals. And then we looked into the gene through the gene expression microarray. We
Hello?
did RNA.
Javed,
Yeah, just done.
sorry to interrupt.
Just
If
done.
you can wind up, yeah, sure.
Yeah,
Thank
done.
you.
So we looked into metabolic genes, we looked into cell cycle DNA expression. So looking into the mechanistic illustration of the studies ultimately indicate that with age, there's a disruption in gut microbiota. And this disruption in gut microbiota ultimately it influences the function of dendritic cells. And then these dendritic cells, they acquire hyperactivation property. And because of this hyperactivation property, they have capacity to even break down the tolerance of T cells, which are undergone central tolerance in the peripheral blood. So this is why we get a lot of autoimmune diseases with old age and other inflammatory diseases. So I'm going to stop here, and I'll be happy to take any questions.
Thank you so much, Javed, for the exciting talk and sharing the animal data linking aging with the gut micro composition and also immune dysfunction. And you have come up with some therapeutic intervention in the form of a species of Lactobacillus plantarum. It's a very good lead. We'll take a few questions. please.
Wonderful talk, sir. So how did you home in on only one species? There might be many others in that particular phylum that you showed. What was the criteria for that?
So the criteria was, although there was upregulation, but what we observed that in Lactobacillus plantarum, it was the maximum decline in the gut of old we observed was Lactobacillus plantarum. And that is the reason ultimately we decided on this.
Out of all the candidates that were there.
Above all the candidates. Yes.
There is one question from here.
This side, chairperson.
Sir, probiotics with anti-aging ability refers as zerobiotics. I hope I'm
Can you speak loudly?
probiotics with a delay in aging. We refer them as a zerobiotics.
Zerobiotics.
Zerobiotics. So with reference to the zerobiotics, because you took only one culture, why not checking multiple strains and looking which is the best one with reference to the gut microbiota modulation as well as immunomodulation and dendritic function also.
See, even though there were a lot of dysregulation in other species as well. So we have chosen over here. Initially, we started using different strains.
Okay.
But what we observed in that, if you take a consortium of bacteria, so they were, reciprocally regulating each other. So you mix particular proportion, and when we injected them, we could not see their retention in gut. Plus, the frequency was frequently changing. So that was the reason we tried to stick to one particular species. And since one Lactobacillus plantarum was giving us a substantial difference, and that was the reason we selected this for population.
Thank you.
Actually, this question crops up again and again. So since you were ultimately targeting the immunological cells and that two dendritic cells which use process antigens, like Bhavtosh once did the complete NGS of Lactobacillus rhamnosus, and there were 23 different types after the full sequencing. And they have motifs which through the different bioinformatic tool, one could find their functions. So picking up antigens or cocktail of antigens to trigger could be one of the other bases to find out. And a lot of these signatures are now available in the literature for most of the probiotic strains. But one of the things which I found very interesting over here, that they could actually stimulate the exhausted T cells. So in the CAR T cell therapy, which are coming up these days, after some time the exhaustion of T cells happen. And there are a lot of strategies which are being put up. Different kind of compounds are given to again bring back those T cells from the exhaustion. So this could be a very good approach to move them. But I don't know whether in those sterile conditions, these could be given as a whole organism, but maybe an approach to look at the antigens or subunits could be also one approach.
Yeah, this is a very interesting question. And definitely, if in such cases, like especially you're talking in context with cancer, instead of giving whole bacteria, if we identify some of the metabolites, so that will be quite interesting. And giving Lactobacillus plantarum, we could observe that there was the cells which underwent exhaustion or the cells which were tolerized. When we cultured with these cells, dendritic cells which were stimulated, we obtained from animals which we injected Lactobacillus plantarum. So they had a capacity to induce the proliferation of tolerized cells or exhausted cells.
So
There's one question from here. Last question,
my
please.
question is the same extension of what Professor Ganguly has asked. So we are doing CAR T cell therapy and as well as bispecific T cell therapy or T cell engaging therapy. And one of the problem which we face is the T cell exhaustion, which happens in these patients, and the CAR T cell therapy or the bispecific therapy that doesn't work. So one of the thing which is coming up is the use of short chain fatty acids, the diet which is rich in short chain fatty acids. And so, you know, I know you have worked on Lactobacillus plantarum, but you think, what is your view on, you know, use of the short chain fatty acid diet, the diet which is rich in this?
Yeah, again, this is a very interesting question. So we are now looking into metabolites. So whether it's short chain fatty acids or some other metabolites, which are important for you know, overcoming exhaustion in T cells. So that will be interesting if you feed these short chain fatty acids or some other metabolites, whether in such cases you can break down tolerance or tolerized cells or exhausted cells. So this has quite potential because in many cases, especially in immunosuppression, even lactobacillus, they start proliferating to an extent that it becomes difficult to control. And another problem we faced was the colonization of lactobacillus in the intestine. So for that was the reason that we have to give regularly. You might have noticed in our experimental design that we give regularly, constantly we give for many days Lactobacillus plantarum. So in that case, even the lactobacillus or any antigen you keep on exposing the immune system, it becomes foreign. So even the tolerized cells, they may get activated. So giving the metabolites may be a very ideal situation. It may be a very judicious approach.
Thank you. We'll take the second speaker.
So it's my pleasure to invite the second speaker, Dr. Anil Kumar, who is a staff scientist level 5, and he is in Gene Regulation Laboratory, National Institute of Immunology. And he is going to speak on towards investigating the anti-cancer role of gut microbiota-derived metabolites. He has published more than 75 in publications in international journals, and he has participated in the development of biosensor, which has now been transferred to the industry. And he also has one of his patents selected for CSIR technology. He's heading the IPR division of the National Institute of Immunology, and he has been granted more than 50 patents. So the stage is all yours, Dr. Thank you.
Thanks for kind introduction. Thanks to Professor Ganguly and Dr. Nirja for inviting me here for this event. So today I am discussing about implication of gut microbial metabolites in the causation and treatment of cancer. Particularly, I will focus on colorectal cancer. So as you all know, we are not alone. We have a lot of bacteria, a lot of fungi in our gut or on our body. And you will be surprised to note that more than 10% metabolites which are flowing in our blood are actually coming from different bacteria, different fungi, short chain fatty acid, as we were discussing. So, and other metabolites like TMO, TMA, all these metabolites are coming from bacterial and fungi origin. So these metabolites are playing important role in human health and diseases. So today we know what kind of bacteria we have, what kind of phyla we have in our gut. but we don't know much about metabolite and functions of metabolites. So last 10 years have been devoted to identify what kind of metabolites are there from different bacteria and fungi. So with the help of mass spectrometry and NMR, we identified a number of metabolites. And today we know more than 200 metabolites, which are quite important. And these metabolites like short chain fatty acids, trimethylamine oxide, trimethylamine oxide, paracrysyl sulfate, indoxyl sulfate, phenyl sulfate, all these metabolites are playing very important role in making us healthy or causing different diseases. If these metabolites are not in physiological range, then there are implications in different diseases of these metabolites. So these metabolites, when they are produced in the gut, they may have a local effect. They can interact with the intestinal cells. They are responsible in maintaining gut barrier function. They can, even if metabolite like colibactin, they can damage DNA. So, and these metabolites may also have a direct systemic effect. They can go to heart, brain, lung, and liver. Okay. So if they go to kidney, metabolites like EPS or PCS, indoxyl sulfate, these are uremic toxins. And these metabolites can interact with albumin, and they can cause chronic kidney disease or other kind of kidney ailments. Many metabolites go to liver for conversion, like trimethylamine. Trimethylamine get converted to trimethylamine oxide, and then they have their physiological functions in human body. So these metabolites have roles in human health and diseases. These metabolites are also important in our immune system, like indole derivatives or short chain fatty acids. These metabolites interact with the different immune cells. These metabolites are responsible for maintaining tight junctions of gut lining, and they are also important in T-reg differentiation or T-cell activation. They interact with different kind of immune cells. So these metabolites are quite important in maintaining our immune system. So my talk is related the link between cancer and microbiota. So cancer has different causation factor, but one factor is biological carcinogen. So according to WHO, more than 15% of cancer diagnosed globally are attributed to biological carcinogens like bacteria and viruses. We all know about Helicobacter pylori, which cause gastric cancer, and Epstein-Barr virus can cause lymphoma. Hepatitis C virus has been implicated in the causation of liver cancer. HPV is well known to cause cervical cancer in women. So in this field of linking cancer with gut microbiota, we are wondering whether some bacteria like Helicobacter pylori may be there to cause colorectal cancer or other kind of cancer. So bacteria or bacterial metabolites are responsible for causing some percentage of cancer in human. So H. pylori, like when we are trying to link bacteria with cancer causation, so this field is quite important, and you can note this when Australian scientist Barry Marshall identified one bacterium, Helicobacter pylori, he got Nobel Prize. So after finding this bacterium, so we are still searching some other bacteria which can cause colorectal cancer or other kind of cancer like pancreatic cancer. So in this field, we are trying to implicate bacteria and bacterial metabolite. And these are the bacteria for colorectal cancer causations like Fusobacterium nucleatum, E. coli, P. plus strain, Enterococcus, S. bovis, Bacteroid fragilis, H. pylori. So all these bacteria have been implicated. Though we don't have sufficient evidence or we don't have concrete proof to prove these bacteria are really causative agents, but like they are secreting some metabolite like colibactin. So colibactin may be responsible for causing colorectal cancer. So here, like we have a lot of bacteria, like E. coli or Fusobacterium or Bacteroides fragilis. They secrete some harmful metabolites like trimethylamine colibactin. And these metabolites can interact with the epithelial cells. They can damage DNA, or they can interact with some signaling pathways. They can make epithelial cell to secrete some cytokines which are inflammatory in nature, and either single event or all these events in a combined way, they can cause inflammation. And finally, colorectal cancer, fully blown cancer can be seen. So far we implicated bacterial metabolite bacteria in the causation of colorectal cancer, but there is a field which is upcoming, which is connecting microbiome with the cancer causation. So microbiome is the collective study of fungi which are inhabiting human gut. So Aspergillus, Penicillium, Fusarium species, they secrete mycotoxins like alpha toxins, and they interact with the epithelial cell. They can damage DNA. They can activate some signaling pathway which are implicated in the cancer causations. So these two fields, microbiome and microbiome, these two fields have been implicated in the causation of colorectal cancer, and not only colorectal cancer, but other cancer like pancreatic cancer or gastric cancer. These bacteria and bacterial metabolites have been implicated recently. And so far, we have evidence for colibactin. Colibactin is a large metabolite which is secreted by E. coli and Klebsiella, which are having PKS island in their genome. And these colibactin, lot of papers are out which prove colibactin can damage DNA, and colibactin is responsible for colorectal cancer. So besides colibactin, we don't have other metabolites with concrete evidence. Okay, there are many metabolites, there are many bacteria which have been implicated, but solid evidences we have for colibactins only for the causation of colorectal cancer. So far we discussed about negative roles of metabolite in the causation of cancer, but some microbial metabolites may have positive sides. They may be implicated in the anti-cancer potentials. So in 2021 landmark paper came, it is published in Science, and this paper showed inosine, which is microbial metabolite, can enhance the efficacy of immune checkpoint blockade therapy. So after this, then many paper came to prove metabolites like short chain fatty acid or other metabolites can be implicated for the treatment of cancer. So like short chain fatty acid, there are many papers which discuss about the anti-cancer potential of butyrate or other kind of short chain fatty acid like acetate, valate. Okay, so these metabolites, short chain fatty acid metabolites can inhibit cancer cell proliferation. They can induce apoptosis, or they have different mechanism to kill the cancer. And these are the possible mechanisms through which bacteria can target tumors. These bacteria or metabolites can do autophagy or apoptosis, or they can make cytotoxic T cell to secrete perforins or granzymes. They can interact with the T regulatory cells. So there are so many mechanisms through which these metabolites or bacteria can act against cancer cell. So in my lab, I'm screening different metabolites, microbial metabolites for their anti-cancer effect. And I studied 4-ethylphenyl sulfate, p-cresol sulfate, indoxyl sulfate for their anti-cancer activity. So 4-ethylphenyl sulfate is a common metabolite now, and from our diet, some gut microbiome can convert dietary component into 4-ethylphenol, and 4-ethylphenol goes to liver, and liver can convert 4-ethylphenol to EPS, 4-ethylphenyl sulfate, and then this metabolite comes again to blood for its physiological functions. So we exposed cancer cell SCT116 and normal cells, and we found Like we took different time point, 24 hour, 48 hours, 72 hours, and we found like cancer cell health is compromised at concentrations of 5 millimolar or 10 millimolar. But when we compared these results with normal cell line, we also did MTT assay on normal cell line, CCD 841, which is normal colonic cell line. And here we can compare damage to normal colonic cells was not much. So ethyl phenyl sulfate selectively killed a cancer cell, but it is not putting damage on normal colonic cells. Then we did different viability assays like ATP count assay, and we saw the same pattern. We saw like at concentrations of 5 millimolar or 10 millimolar, cells are compromised. We did then cellular imaging. We saw cellular morphology is distorted at 5 millimolar or 10 millimolar concentrations. Then colonogenic assays we performed, and we saw at concentrations beyond 2.5 millimolar, colonogenic ability of the cells were compromised. We checked apoptosis, we checked like Roche generations, and we saw like beyond 5 millimolar or 2.5 millimolar, we have compromisations on cell health, cancer cell health. And we also checked through Western blot Bex and BCL2 ratio, and we saw beyond 2.5 millimolar. This ratio was increasing, which is suggesting like apoptosis is happening. We also studied cell cycle arrest and like GTM cell cycle arrest was seen. Then we checked peak cresol sulfate product effect of peak cresol sulfate on cancer cells. And here we saw same pattern. Like we used SCT116 cell line and normal colonic cell line, and we saw like at 5 millimolar or 10 millimolar concentrations, all these metabolite EPS or PCS, they are damaging cancer cells, but they are not damaging normal colonic cell. So this effect is quite selective, and like cellular imaging and other assays we studied, we saw same pattern when we did ATP count assay or when we did other LDH assay, which is about membrane integrity. And colonogenic assays we also performed for PCS, and we saw like at 5 millimolar or 10 millimolar concentrations, cells are compromised. We did cell cycle analysis, and then apoptosis and Roche assays we performed for PCS, and we saw similar result as we saw in the case of EPS. Then third metabolite I selected, that is indoxyl sulfate. Indoxyl sulfate, if we have dietary tryptophan, so some bacteria can convert dietary tryptophan into indole, and these indoles can go to blood and from blood to liver. And on liver, indoxyl can be converted to indoxyl sulfate through the action of sulfotransferase. And then again, indoxyl sulfate comes to blood for its physiological functions. So why we checked indoxyl sulfate for its anti-cancer activity? Because many drugs which are in clinical practice are actually indole derivatives. Drugs like nintedanib or sunitinib or vincristine, vinblastine, all these drugs are indole derivatives. Indoxyl sulfate was also indole derivative, but it was made by bacteria. It is gut microbiota derived metabolite, and there was no paper or no evidence for its anti-cancer activity. So we thought we should check its anti-cancer activity. This was the premise. So here we did the same thing, but in this case, we took two cell lines, SCT116 and ST29. And we saw beyond 2.5 millimolar concentration. So cell health was compromised. Then we did ATP count assay and we saw same result at 5 millimolar or 10 millimolar concentration. Cell health was compromised. We did LDH assays, we saw same result, and then we did apoptosis and we saw like beyond 1.25 millimolar concentration, apoptosis is increasing as we are increasing as we are increasing the concentration of indoxyl sulfate. Then we did all these assays as we did in the previous with previous metabolites. Then we extended our study to animals and we saw whether indoxyl sulfate is safe to use. If we consider indoxyl sulfate for further application to use it as a drug, so we gave indoxyl sulfate to animals and we saw like indoxyl sulfate is not damaging intestine, or like there was no difference in colon length or spleen length. So indoxyl sulfate is quite safe, and we gave indoxyl sulfate at high concentrations like 50 millimolar intrarectally and intraperitoneally. We did the histology study, and we see when we compared with the vehicle control. So colon tissue and rectum tissue, we didn't see inflammation or any damage to the tissue after exposing of indoxyl sulfate. We also checked inflammatory cytokines after giving indoxyl sulfate, and we saw like in cytokine level like IL-17 or IL-1 beta or TNF alpha, these kind of cytokines we checked, and we saw like the difference is not significant with the control. So there was no inflammation after giving indoxyl sulfate to animals. So this is the conclusion. Our results showed indoxyl sulfate is responsible, not indoxyl sulfate, but other metabolites. They are responsible for inducing apoptosis, cell cycle arrest, loss of membrane integrity. And all these metabolites are killing cancer cell in a very selective way. So they are killing cancer cells, but they are not harming normal colonic cell, or they are not harming colon part of animal
time. So I think you have talked about the different metabolites which have anti-cancer role, but in the gut homeostasis, when it is there, do you think it is the one metabolite, or it is the imbalance of the metabolites which causes cancer versus which are anti-cancers, which are, leads to the cancer formation? So I believe as a clinician that it is mostly the imbalance of the anti-cancer versus cancer-causing metabolites. What do you think about that?
Okay. So some metabolites like colibactin. So it is not about balance, but if colibactin is secreting, if some bacteria like PKS plus E. coli or PKS plus Klebsiella, if these bacteria are present and they are secreting colibactin, whether colibactin is secreted in less amount or large amount, if colibactin is there, then it is sure these metabolites will damage DNA, and this DNA damage or inflammation can cause colorectal cancer in long term.
Okay, the talk is open for discussion. Yes, sir.
So in continuation of what you said, see, you mentioned both the bacteria which are pro-cancer, like the metabolites are promote cancer, versus those metabolites you later showed in your study which act as anti-cancer. Now, these two populations in the gut would again depend upon the diet. So what are the dietary linkages to the different populations will actually ultimately translate into effective interventions. While the metabolite as therapeutic approach is a different one, the linkage with diet is also equally important if you want to prevent this.
Okay. So diet is one factor, but infection of these bacteria is other factor. Like Salmonella typhi, if we get infection of Salmonella typhi or H. pylori, okay. So some bacterial or like PKS plus E. coli strain or Klebsiella. So diet can modulate the bacterial population, but infection is also one factor. We can contract these bacteria from other person or from environment. And if we are infected by these bacteria, then they can secrete these genotoxic metabolite or genotoxins.
I will take what Dr. Malhotra was saying, we'll take it further. Since yesterday, we were looking at different papers which were presented about the microbiome population in the colon cancers. And there were distinct signatures of different types. So colibactin can cause cell damage, DNA damage in cells, but it can also eliminate the other bacterial community. It is a kind of a bacteriocene. So it might eliminate some of the other beneficial organisms and ultimately change the environment. So it will be interesting to see the microbiome population in the animals or subjects where the colibactin, enhanced colibactin is being produced. So the disturbance of the communities or environment in colorectal cancer could be equally important.
You are right, sir. So there are many comparison reports between healthy people and cancer patient. Their microbiota is different. You are very right. Like these metabolites can alter the microbial population. You are right.
I have an extension of Dr. Shashikaran's question. So basically, indoles, phenols, and cresols are produced as a result of bacterial metabolism of nitrogenous food. So it's proteins which are not absorbed in the small intestine, which escape into the colon, which lead to these. And sulfation of these indoles, phenols, and cresols happens both in colonic epithelium and in the hepatocyte. It's actually a mechanism by which the body detoxifies this and excretes it either in bile or in the urine. So I think what you have shown is probably that indole sulfates can have an anti-cancer effect. You're trying to use that as a drug. But in nature, what happens might be that these indoles, phenols and cresols may actually be carcinogenic in a different way. It's possible.
Yeah. Actually, these indoxyl sulfate and paracrysyl sulfate, ethyl phenyl sulfate, all these are uremic toxins. Okay. So these metabolites are quite negative in their nature, but they also have positive sides. Okay. We studied their anti-cancer activity. So they have dual functions. We don't know what is the mechanism, but we found anti-cancer activity of these metabolites.
My question is that I'm a microbiologist and we know E. coli and Klebsiella are normal commensals of colon, right? So as you mentioned, there is an island of genes which is responsible for colibactin production. So what percentage of these bacteria carry these genes? Because they are normally present in the intestine.
Okay. So not all E. coli have PKS islands in their genomes, but some specific strain of E. coli. So they are not present in healthy people. They are present in people who are having colorectal cancer. So in deceased person, they can be found, but I don't think they are present in normal. So when we compare metagenomic analysis, healthy versus cancer patients, so they are not present in healthy people.
Can we identify their source in the environment, how these people are acquiring?
Okay.
Can we work on that?
Okay, I don't know that how E. coli PKS
I think
plus.
further discussions during tea time or the lunch time. Please to stick to the time. Thank you so much, Dr. Anil Kumar, for your interesting presentation. We'll take up the last presentation for the session. Dr. Tarini Shankar Ghosh. He's assistant professor in computational biology in Indraprasth Institute of Information Technology, Delhi. He has published more than 50 research papers, including Nature Medicine, Nature Ecology, Evolution, Gut Gastroenterology, and Nature Reviews, etc. He's a recipient of Yakult Young Investigator Award in 2014 and the Torrent ISHR Young Investigator Award in 2017. He is author to multiple US EU-based patents, primarily pertaining to efficient metagenome informatics algorithms and identification of putative therapeutic novel microbial consortia. Please, Dr. Ghosh, your time is for about 20 minutes. Thank you. We'll appreciate if you restrict to the time.
Thank you, ma'am. I will. So first of all, thank you to Professor Ganguly and Dr. Nija for giving me this opportunity to present some of my work here. So, like I mentioned, like 2014, I was here presenting as a young investigator, right in my PhD days. So it is good to be back here and present some of my recent work that I've been doing now. So basically, like my background is a bit different. So I'm basically a computational biologist who works with data, and my focus area is human microbiome research. So basically what I do is I investigate the microbial communities within different body sites using a combination of different statistical analysis, data analysis techniques. So like I mentioned, so what I do is like there has been a recent deluge of microbiome sequence data, and this deluge has given us the opportunity to actually use data science-based strategies to answer questions regarding the microbiome on a global scale. And my group, which is like a little more than 2 years, so we work on development of algorithms, methods, as well as we aim to identify and formulate microbial consortia that can be tested, or strain collection that can be tested for various purposes and utilized on a global level. So, this is what the data collection that we have been able to build. So the first thing that we need for our research is data. So over the past couple of years, we have built a collection of close to 270,000 microbiome data sets in our group. It has around 150,000 processed data sets by now, where we have also created the metadata, and they together include around 863 studies. So we have primarily focused on the gut, and we are now expanding it to other body environments as well. And for each of these data sets, what we have is we have generated the taxonomic and functional profiles, their strain variation profiles, as well as the growth rate profiles for some of the major microbes. And at the same time, we have also collected microbiome-derived genomes from the large-scale MAG repositories, as well as from other reference sequence databases. And these include around 70,000 high-quality genomes from around 4,700 species. And we have their full functional annotation using different schema. And then what we use is we use a combination of different kinds of statistical machine learning-based strategies, time series analysis, genome scale, protein scale-based language models, and meta-analysis approaches to answer different kinds of questions. And so one of what we do is some of my previous and current thesis, I work on the area of understanding the interaction between gut microbiome aging, between healthy aging and accelerated aging, as well as in looking into the age-specific microbiome signatures. Some of my work has been on understanding the gut microbiome as a modulator of therapeutic interventions, especially nutrient diet. Looking into the variation of the gut microbiome across populations and lifestyles, especially with lifestyle changes and especially for ethnic minorities. And at the same time, we have also worked on multiple diseases and identifying consistent disease alterations, consistent microbiome alterations in different kinds of diseases. And after coming here, we one of the other sub areas that I've been expanding into is like people have talked about the gut microbiome being very variable across populations and across different kinds of individuals. But what we are striving to do in our group is to actually identify the common signatures or the universal signatures of health, or the universal hallmarks of health in the gut microbiome across different populations, because that is what is translatable. So, some of the other things that we do in our group is to understand the remote interactions between different kinds of body sites. We are also building up matched curated data sets which have metabolome as well as microbiome information, and we are utilizing it to to basically understand the microbiome and metabolite panels or associated, like the panels or metabolites associated with different kinds of cardiometabolic and neurodegenerative diseases. At the same time, so what we are trying to also build is not only not taxa specific, but function derived microbiome predictive algorithms, which actually give you disease risk for various diseases, as well as for the first time adding the component of explainability in microbiome-based disease classification. And the last thing that we do is actually to build predictive algorithms for personalized response to different kinds of probiotic therapies. Now, the last theme is something that I will be presenting here, and we'll be presenting some of the very preliminary results of some of one of our recent studies that we have been doing in this regard. And the study basically is to identify the non-bifidobacterial elements of the baseline gut microbiota that affect the abundance of bifidobacterial species in the gut. Now, Professor Javed mentioned that one of the main reasons that we have to give repeated probiotic supplementations is many of this time the probiotic it does not colonize in the first go, and the effect is even more when it comes to humans rather than in mouse studies. So that is the reason that forms the basis of the studies. Now, bifidobacterium as a probiotic consortium, we all know what bifidobacterium is and what bifidobacterium is important for. So it is one of the well-known health-associated gut microbial lineages. Bifidobacterium has been known to facilitate nutrient absorption, promote brain health. It has antivirus, anti-infective activity, anti-cancer activity as well. It regulates the immune system, the host immune system. It has role in anti-inflammation, as well as in for modulating obesity and cardiometabolic health. And even now, I saw in recent paper where bifidobacterium supplementation has been used to treat functional dyspepsia. So we know what bifidobacterium is and why it is so important. And this just shows, so this was done by one of my students. So it just shows so it just basically gives the number of successful randomized controlled trials per year with different kinds of some of the major probiotic lineages. And as you can see over here from 2014 to 2023, you see that in 2014, the lactobacillus-based trials were more predominant. But over the year, what has happened is we have an increase in the number of trials with bifidobacterium species coming up. And that actually shows the role of these species in different kinds of probiotic therapies. However, the problem is, and what has been repeatedly also cited in other papers, and what we have also shown in this in our analysis article in the British Medical Journal, where we did a thorough review of 214 intervention trials, is that not all trials are equally successful. Not all who are administered the probiotic respond equally well, and show variability in colonization. And these colonization patterns are dependent on the personalized factors, including the baseline gut microbiome. And so that is the reason here what we try to do is, because we have the data pertaining to the different kinds of bifidobacterial species and from the gut microbiome, we decided to see, can we actually identify elements of the gut microbiome that are associated with abundance of different kinds of bifidobacteria. So basically, we are trying to come up with some information which can be used to stratify individuals during probiotic therapy so that we can decide who is going to respond fast, who is going to respond late, or can we actually identify individuals in this regard so we can adjust the dosage and the duration of the interventions for a response in terms of colonization as well as for the host part. So here, the overall view of my talk, and I'll be presenting a very small part of what we have done in this study. So the first part, I will be doing some overview of the detection of the different major bifidobacterium species that we got across different population types, age groups, and depending on the different kinds of microbiome profiling strategies. Secondly, I will talk about the actual identification of some of the non-bifidobacterial gut microbiome members that associated with the abundance of different bifidobacteria, and our derivation of species-specific bifidobacterium scores. Thirdly, at the end, I will present before you a consistent bifidobacterium associated taxa panel and its validation in an independent intervention cohort. So first, coming to the data sets. So here in this study, this was actually performed like in the beginning of our data curation, like within the first 3 months. So by that time, we had around 46,000 gut microbiome data sets collection done by our group from 116 cohorts. So as you can see over here, we have both equally 16S as well as WGS samples. And in terms of the number of samples on the different age categories, so we have a more or less even representation from infants, adults, and seniors. And the only problem that we had, and there's a general problem with global microbiome data, is a bias towards cohort from industrialized and urbanized countries. So that is what we see here. So we profiled the gut microbiome populations across these 46,000 microbiomes, and also we looked into specifically the bifidobacteria. So overall, we detected around 46 bifidobacterial species that were across these 116 cohorts, and we identified 8 bifidobacterial species that were detected with a relative abundance of at least 0.0001 in at least 5% of the gut microbiomes. So this list is actually presented below. We have 8 major bifidobacteria: Bifidobacterium longum, adolescentis, Catenatum, Pseudocatenatum, brevi, Bifidum, Dentium, and Animalis. Animalis is one of the most widely used probiotics, if you can know, as well as along with longum. So this is the overall pattern that we saw. So as you can see over here, that there are certain clear-cut trends. Like if you look into the global microbiome, longum and adolescentis actually predominate. We also see for the infants, we have a higher detection of bifidum and brevi. If you look into the another pattern, you see a very clear-cut difference. If you look into this pattern, you see that in terms of the sequencing, majority of the 16S sequences, we see lesser number of bifidobacterium. So that I will show you in the next slide. And we also see certain clustering of the study cohorts based on their population type. So this actually gave us the inclination to actually investigate this further. So what we can see over here, so this is, as you can see, these PCA plots, every point represents a study cohort, and the plots are plotted based on their abundance patterns of different kinds of bifidobacteria. Now, as you can see, we see a significant difference between the bifidobacterium detection pattern in the infants and adults, between the WGS study cohorts and the 16S study cohorts, and as well as to some extent between the different kinds of population types. In terms of the different species, we see that different species show different patterns. There are some species like brevi and bifidum, which actually decrease with age, like from infants to the seniors. Then we have some species which actually increase with age, like animalis and adolescentis. And then we have some other species which are like decreasing in general, which decrease with the rural and urban rural population, but are higher in the urban and the mixed population. And most importantly, the pattern we see is the detection of different bifidobacteria is actually confounded by the sequencing strategy. In 16S, we have a significantly low bifidobacterium detection for multiple bifidobacteria. So 16S, there is some issue in the 16S profiling of bifidobacteria that has been noticed in previous primer-based studies as well. Now, despite this, one of the most important things that we observed here is the association of non-bifidobacterial diversity with the bifidobacterium detection rate. So the last panel that you see here is basically show the significance of the association of the non-bifidobacterial community population with the overall detection rate of bifidobacterium in the different studies. And you can see over here that at least 70% of the data set, the non-bifidobacterial community population was significantly associated with the number of bifidobacteria that were detected. And this actually gave us the motivation to go further and investigate the individual data sets. So what are the non-bifidobacterial members that consistently associate with different bifidobacterium species? And to do that, what we did is we used a combination of machine learning and correlation analysis. So this is a very simple analysis. So what we do is we take every study. So we had around, for every species, we had around a different number of studies ranging from 80 to 97. And for each of these species, we had the non-bifidobacterial abundance profile, and then we had the abundance of the corresponding bifidobacterium species X. Now, because the number of features are so many, so sometimes we have to do kind of a feature reduction to increase the statistical power of test. And for that, what we did is we used machine learning-based models, random forest-based models to identify the most important features for every study set. And then using those study-specific important features, we build a list of consistent features. And these consistent features then we use to identify their association with a given bifidobacterium species using the Kendall-Tau correlation approach. Now this process, like we have seen over here, this process was adapted for each of the 8 bifidobacterium species. And because there is a difference between the WGS and 16S, this process was performed separately for the WGS and 16S cohorts. And we also performed separate investigations for the infant and the adult. So they were for every species, we had actually 4 different association patterns that we computed. Now, this is for one example where you have computed the association pattern of bifidobacterium adolescentis in the WGS course and only for the adult and senior populations. We do see that there's some very strong consistency. You see the blue boxes over here. You can see Collinsella, Faecalibacterium, Coprococcus. Collinsella is another member. Actinobacteria, we see a lot of good bacteria like Faecalibacterium, Coprococcus, Eubacterium rectalis, Akkermansia mucinifila that are in these blue boxes. That show consistent positive associations with Bifidobacterium adolescentis. At the same time, we also see some of these members. If you look into the member, Enterocloster, Citrone Enterocloster, Bolti, and Clostridium symbiosum, all of these have been known to be associated with diabetes, and they have been they are basically in the group of Pathobiont Clostridium, which show a negative association. So we got these consistent patterns, and which can be seen here. The top line plot actually shows the pattern of association. So blue means in how many study cohorts the species was positively associated. Red means in how many study cohorts it was negatively associated. So we got those. We saw those kind of consistent patterns. And then we decided to actually grade the species based on the consistency of this pattern. So the pattern of association could then be used to build. So we decided to build Bifidobacterium specific association scores. Now these scores were basically Bifidobacterium species specific, separate scores for every species, and separate scores for different microbiome profiling strategies, and computed separately for adult and senior the infant microbiomes. So what we did over here is this line plot is what I showed you yesterday. So we built a very simple kind of a score. This is like, for example, the score for taxa I for association with Bifidobacterium adolescentis in WGS and this microbiome is essentially the number on the blue line. That is the number of cohorts where the WGS is significantly positively associated, and minus the number of WGS cohorts where there is a significant negative association, that is the red line, divided by the total number of WGS cohorts that we are investigating. So this is a very simple score. And when we did that, this is for one particular aspect for the WGS cohorts that we see over here. We see, so this we did for all the species, like you can see, and we see that there are some taxa over here which showed consistent positive association with multiple Bifidobacteria. You can see these blue boxes. This has a 23, this 19 plus 23 taxa panel. So it's a 43 taxa panel that we detected that showed consistent positive associations with multiple Bifidobacteria. And if you look into the list, this is the list. So there is one group which shows the association with all the 8 Collinsella aerophaciens, Clostridium leptum. Then we have a lot of Streptococcus, Collinsella massilensis, violent. So it's a mixture of Collinsella, Streptococcus, and some of the oral bacteria. There's a second list, as you can see over here, another group which has a lot of health-associated gut bacteria, ranging from Faecalibacterium to Fusicatinibacter to Agathobaculum, a lot of butyrate producers over here, and along with Akkermansia mucinifila. So these are the 2 groups that we observed over there. And this is the constituent of the 43 bacteria that showed the most consistent association with all or most Bifidobacterium populations. And so this 43 panel, then what we did was we thought that, okay, we have identified a panel, and can we actually utilize this panel to predict something in intervention cohorts? So for that, what we did was, if you remember this first study where they showed that the Bifidobacterium response is associated with the resident gut microbiome, but they could identify just Collinsella to be significant associated. There was no other association that they were reported. So we see that if we could actually utilize this 43 taxa panel to predict the post-treatment response in these individuals. So the x-axis basically shows the post-treatment Bifidobacterium abundance of these individuals who were actually given the Bifidobacterium longum supplementation. And the y-axis basically is a very simple measure where we actually computed took only this 43, and we computed a mean rank scale abundance of the Bifidobacterium positive taxa. And as you can see over here, this score was computed at baseline. The y-axis scores are computed at the baseline, and the abundance that we see are at the post-treatment time point. And as you can see over here, there seems to be a very strong correlation. So we are now extending it to other intervention trials as well. And we have investigated around 6 to 7 trials, and in around 5 or 6 of them, we see that the post-treatment abundance is predicted, is significantly predicted using the pre-treatment abundance of these bacteria. So this is just one part of the study. So we have investigated additional things like these ranking scores we have seen are correlated between WGS and 16S studies. We see a lot of similarity in the ranking patterns between the infant and the adult gut microbiomes, albeit with some kind of variations. And we have also extended these investigations to the strain level. And we see that, especially in the case of Bifidobacterium longum, where we see that there are 2 major clades of Bifidobacterium longum, and you can actually predict the clade. One clade is associated with a lot of antitoxin systems, a lot of transporters, permeases, and integrases, as well as a lot of antimicrobial secretion peptides, which is associated with certain kind of bacteria. Then we see a lot of carbohydrate active, another clade enriched for a lot of carbohydrate active enzymes, which are associated with a second group of bacteria. So what we summarize in this very small part of the study that I was presenting of a bigger analysis is the bifidobacterium component of the microbiome varies according to age, lifestyle, urbanization status. We see that the bifidobacterium detection pattern is significantly impacted by the microbiome profiling strategy. Some of the known probiotics like animalis is not often detected in the human gut. It is the other way, the longum and adolescentis were prominent. The baseline non-bifidobacterial components significantly impact the bifidobacterium component of the gut microbiome. And we see a set of 43 taxa and 19 and the subset of 19 taxa, we show 43 taxa which show at least with 6. The 19 taxa are the one that are showed with all 8. We show consistent positive associations with the majority of the bifidobacterium taxa abundances. And these 343 taxa can also be used to predict future colonization pattern of bifidobacteria in probiotic interventions. So we have more validations going on in these lines. So with that, I would like to end my talk over here. I would like to thank my group. So we don't have 2 more members here. I would like to thank the Department of Computational Biology, IIIT Delhi, for the support. And I would like to also thank the funding agencies who were instrumental in driving this research, especially for creating the data collection we are working on. So thank you. And then I would like to take questions.
Thank you so much for appreciating the timeline. We'll take a few questions now.
So in this population, how you took into consideration epigenetic changes?
So we did not consider the epigenetic changes here because this was all public data. So the epigenetic factors were not considered here. So we took the data sets from, we had the metadata pertaining to the things that I mentioned. So whatever metadata we could collect, and that is what we worked on.
That was the question, that while collecting data,
yeah,
so did you took in consideration epigenetic changes?
No, not because epigenetic, there are very few data sets where they have profiled the epigenetics and the microbiome. So that's what we are now working on to actually get to those kind of, especially in aging scenario, those data sets are going to be very important. But I think there are like a couple of data sets available which have the epigenome as well as the metagenome being sequenced.
You have thrown another turbulence in the system that 16S and WGS data might vary.
Yes.
Both in the species, the strain variations as well as the abundance. So,
yes, so that is exactly what we see here also.
What I'm worried about that the interpretations in the literature which occurs using the different techniques might create data which may not be comparable.
Yes, so that is exactly what we see here, sir. Like 5 or 6 bifidobacterium, you could see that their abundance in the 6ness is significantly lower than a WGS.
Where have you published this data?
So we have not published it. This is still an ongoing story.
Publish it quickly.
Yeah.
Any other question? So I have just general observation. Can I just express it here? I heard a lot of presentation yesterday and today also. I think there is too much of emphasis on one species or one gender or one metabolite. But the gut system is very complex. It is an ecosystem.
Yes,
itself.
yes.
There's pH. We don't know. There's an aging. There could be some other factors like diabetes.
Yes.
Taking everything in isolation doesn't solve the problem. Since you are an AI machine learning person.
Yes, so that is exactly.
You can put so many things, a real life situation in the gut and create, a gut-like situation, putting all these.
Yeah, so exactly that is what we are doing now. So what we are doing now is, so in this investigation, the next stage, we have the age, we have the gender, and we have the disease status also. And for some of these data sets, we have the diet as well.
Diet is very, so many complex factors are involved. Taking one, it is good for publishing papers and getting grants.
Yeah, so that is what we are.
The granting agency would ask, or they will ask themselves, what have I got?
Yes.
By funding so many projects.
Yes. So
you have to think, the youngsters especially have to think about.
Yes. So the next stage, that is exactly what we are doing. So we are putting this age. So we cannot get all the metadata because we are still working on publicly available data sets. But for some of these data sets, we have the matched microbiome as well as diet information. So we are putting in the diet information, disease information, the age information. So whatever information we can get along with this 43 taxa panel and trying to check if we can actually predict their efficacy during interventions.
Yeah, Dr. Ganguly, in this connection, even, this consortium they are developing for specific stomach, this thing. A consortium and capsule writing doesn't solve the problem until unless you have somebody who can give you those capsules in GLP condition. And then, you test it in animal model or some other model and go ahead with humans. I mean, sometimes it worries me,
Actually,
because we have struggled
this-
throughout our life on those issues,
Actually-
GLP, GMP, and
actually, this is a very unresolved issue. Like we are just taking a whole bacterium, but yesterday you saw the Japanese paper that LCS has the LCP1.
yes,
and
LCS was a
so-
wonderful
so-
thing.
so-
I was
so
impressed.
These days, like with the plantarum,
Mm.
there was a paper which came in Nature that the early infant septicemia was handled by the plantarum species. And plantarum also, but what was very worrying, which was presented just before this, where you come in, that even the bifidobacterium has so many signatures, long arm, baby, there are so many bifidobacteria.
Speciation is there, wide range.
And it's changing in infants, it's changing in senior adults, changing in senior. This actually, if you really ask me, we have thrown up a huge number of questions without answers.
Yes.
And these are up to the
Yeah,
future generations
yes.
to find out the answer. But this is the beauty of such deliberation that you may not get an answer so very easily, but throw as many questions as possible to the people for the future investigators and let them find out. But communities are important, like VSL3 is one of the products which is a community of about 6 and one fungi. So they work in a tandem, they work in a collaboration. So I don't have answer to these, but individual strains are also important for individual functions. That is very clear. Since yesterday there were several such papers where if you are like there was one where of the GG, Rhamnosus GG, it had a specific function in the mother-child, which Patricia yesterday presented.
Yes.
So
MDR genes in
so
Gujarat
it
women
was
and
very-
in milk. Yes. Sir, I was just wondering if, we can make a compendium of questions after such wonderful conference, 2-day discussion, and then say how many of these can be resolved, because your society, your foundation is very profound. It's almost matured now. And then, we can pose questions to, in a very specific manner to funding department end to end. Sir, one more point, can I just take half a minute?
Yes.
I think that was the first presentation made by Dr. Kadri from Bangladesh. She mentioned about that, this immune response to oral cholera vaccine, whether it is killed or live, is impacted by microbiome composition. She mentioned about varying response from adult to different age group. And then she said the Sanofi has stopped manufacturing Shanpol. I was literally touched. She said, we want cholera vaccine. So I don't know whether a foundation like yours can take up such questions, because we in India may be capable of doing, an alternative, vaccine. I do not know why Sanofi closed it. She mentioned about it, if anyone remembers it, So, because it is taken over. Shanta has been taken over by Sanofi.
was taken over by Sanofi.
And it's not a profitable.
And they didn't see money in this.
Yes, yes.
And that's why they closed it. But the UBio of the South Korea has come up with a version, and they're much better than the Shancol.
Okay.
and you were handling all of this.
Yeah,
So
that's
you are guilty of all the things which you have done.
I'm not guilty. I don't want to open my mouth, but I thought that we have an opportunity in places like that. Thank you so much. And thanks for all the speakers.
Thank you.
Congratulations, man.
I now request our chairpersons to felicitate the keynote speaker. I request Professor Bindu De to felicitate Dr. Anil Kumar. Yes, can we have a huge round of applause? Thank you. I request Professor Pankaj Malhotra to felicitate Professor Javed N. Agriwala and Dr. Tarini Shankar Ghosh. Can we have a huge round of applause for our speakers? Let's have a huge round of applause. Let's have a huge round of applause once again. May I request Dr. Kentaro Shimizu to felicitate our chairperson, Professor Bindu De. Let's have a huge round of applause. Dr. Kentaro Shimizu. And I request Dr. Osamu Chonan to felicitate our chairperson, Professor Pankaj Malhotra. Let's have a huge round of applause. Meanwhile, I request all of you to fill up the post-symposium survey form and hand it over to the girls at the registration counter. Let's have a huge round of applause for our chairperson. Thank you. Moving on to the next session on showcasing microbiome studies from PGIMER Chandigarh, I have the pleasure of inviting the chairpersons, Professor Neelam Taneja, Professor and Head, Department of Medical Microbiology, Postgraduate Institute of Medical Education and Research, Chandigarh, India, and Professor Usha Datta, Professor Gastroenterology, Postgraduate Institute of Medical Education and Research, PGIMER, Chandigarh, India, over these sessions. Can we have a huge round of applause for our chairperson?
So good morning, all of you. So we come to this exciting session. The session is entitled Showcasing PGI Studies. So first thing I'd like to tell you, this is a work in progress. So these are not complete works, and these presentations short, 12 minutes each, and we can have a discussion at the end of the session. So first, I would like to invite Professor Usha Datta, and her title of her talk is Mucosa-Associated Gut Microbiome and its Relevance.
Nice, this whole meeting. Thank you. So what I thought is I'll shift gears from fecal associated microbiome. Most of the studies are using fecal flora, and here we will shift to mucosa associated microbiome. Fecal microbiome analysis has been the most common specimen studied all around the globe, and it assesses the global profile of the gut lumen. It is easy to obtain, but does not need any intervention. But the mucosa-associated microbiome is there at the level of the tissue and is driving the tissue inflammation and injury. It has a specific signature for a person, and it can also help us understand the focal dysbiosis. Gut has a huge surface area, and you would not believe it is as good as high as half of a badminton court. So the fecal bacteria actually resembles the luminal microflora and not the mucosa-associated microflora. And the stool fails to capture the mucosa-associated microflora because this is adherent to the mucus layer at the level of the mucosa. So as I said, the gut has a very huge surface area, as high as 32 square meters. And most of the surface area is in the small bowel, but small bowel has high transit and has good peristalsis. So very little mucosa-associated microbiota stay adherent to that, unless it's a focal area of biofilm or dysbiosis. However, the colon has several areas where, especially the cecum and the upper ascending colon, these areas have a lot of adherent bacteria and local biofilms which drive focal dysbiosis and inflammation. Here in this cartoon, we can see that this is the luminal microbiota. This is the one which gets shed in the stool. And the one which is adherent to the mucus layer, the one which is very closely adherent, and this colon has 2 layers of mucous membrane. And this is what is closely associated to the epithelial cells and drives the mucosal inflammation and immunity. So it is a distinct bacterial composition compared to the gut lumen. It impacts barrier function. It interacts with the immune system. And the mucosa-associated microbiota differs from the fecal and the luminal microbiota. There are several studies which have linked inflammatory bowel disease, irritable bowel syndrome, colorectal cancer, and the mucosa-associated microbiota. So we embarked on assessing the colonic mucosa-associated microbiota in IBD, duodenal mucosal-associated microbiota in diabetes, and the duodenal mucosa-associated microbiota in patients with gallstone disease. So here the duodenum is the seat where it drives the enteroinsular signaling. India is increasingly becoming the diabetic capital of the world. And I wanted to see what was happening in the duodenum in these patients. Because when I was in Canada for a fellowship, I realized that the gut looked very healthy. When we were doing endoscopy, the duodenum looked very healthy. But when I came to India, I found that the duodenum doesn't look healthy. So that is why I thought whether there is a link between these two. And there are studies which have shown that duodenal sleeves help in resolving diabetes and improving the gut glucose metabolism. And studies which have shown that if you resurface the duodenal epithelium, again, the diabetes comes under control. So another study which showed that if you exclude the duodenum, again, the diabetes comes under control. So these are all studies which have shown that the duodenum plays an important role in diabetes, in the enteroinsular signaling. So what interested me is to see whether the microbiota there is different or not.
So duodenum is an area where a lot of physiological activities are happening. Also, the duodenum is a place where the oral microbiome comes here, and in the stomach you have acid, and the duodenum you don't have any acid. So this is the place first the body is interacting with the external microbiota. And this is a place you have a lot of metabolic actions going on, and this is the place for the enteroinsular signaling also. So this is what interested us in studying this area. So we did a study, a pilot proof of concept study between patients who are hyperglycemic and those who are normoglycemic in PGI as well as in Assam. And we did find that we classified patients as hyperglycemic and normoglycemic. And we found we took a duodenal biopsy and did a 16S sequencing on that. And we also did their fasting blood sample, also studied their stool microbiota. And what we found very interestingly, that patients with hyperglycemia had higher bacterial content in the duodenal mucosa compared to normoglycemics. And in all fasting blood sugar, postprandial blood sugar, and HbA1c, whenever it was higher, it had a higher duodenal microbiota in terms of quantitatively. Qualitatively, duodenum had more pathobionts in those who were hyperglycemic and less of the beneficial bacteria, and we had less of the Aracamencia musciphagia. Also, interestingly, we see the duodenal flora helped us differentiate diabetics from non-diabetics in contrast to the stool flora, and thus again emphasizing the role of mucosa-associated microbiome. At the phylum level, we found that there was more proteobacteria in the duodenum, which should not be there, and compared to the stool, and this was different in those with hyperglycemia versus the normoglycemics. We also found that patients who are having the biopsy samples more often had a different profile than the stool sample. We also looked at the duodenal saturation with oxygen saturation using a T-stat method, and we found that those with diabetes had a lower saturation than those without diabetes. And we also found that the duodenal mucosa was more often permeable compared to those who without permeability. So for want of time, we will not go into all aspects of it. All that we would like to summarize here in this study, that the patients who had hyperglycemia had more often pathobionts, less often the beneficial bacteria, excess of quantitative bacteria in the duodenum, and had decreased duodenal saturation probes showing that it had lesser perfusion and lower inflammatory markers. So moving on to the next component, that is inflammatory bowel disease, we shifted gears and wanted to look at Candida in the biopsy culture as something which could be driving the inflammation in patients with ulcerative colitis. So we thought that mucosa, whenever there's a break, Candida colonizes secondarily and can increase the local inflammation and delay the healing and worsen severity of IBD. And we did a study in 96 patients with ulcerative colitis and compared it to 20 patients with irritable bowel syndrome. And we looked at endoscopy. We picked up on cytology and biopsy culture, as well as histology and the Robert histological index to quantitate the degree of severity of histological inflammation. And what did we find? We found a large patients where we had confluent Candida growth, and we also found on past stain Candida, budding Candida present in the mucosa. We know that Candida can colonize the gut, and it may be just a commensal there, but however, we will show how there was excess of Candida in patients with IBD. So what we found on stool culture, 57% had evidence of Candida. Brush cytology had more often Candida in patients with ulcerative colitis. The blue bars represent the ulcerative colitis. The brushing cultures also more often picked up Candida, and the biopsy cultures also more often picked up Candida. And it was the Candida tropicalis and albicans were the dominant species, and the non-albicans species was higher in IBD compared to the controls. More the severity of IBD, more often did we find confluent growth of Candida. So Candida can be present as a part of normal commensal. I'm here talking about more confluent growth and more amount of Candida, which may be the driver. And what we found was it was not just Candida colonization, there was high beta-D glucan levels in patients with IBD. That is suggestive that there is an invasion and interaction with the gut barrier and immune system. Those with biopsy culture positivity had higher clinical and endoscopic activity, higher histological activity, and more often had confluent growth. And those with confluent growth had higher beta-D glucan levels. So here you can see that confident growth on the biopsy culture yielded higher beta-D glucan levels, indicating that with increasing amount of candida, there's more likely invasion. There are some proof of concept, there are just some anecdotal reports that antifungals may have a benefit in IBD. So we explored that given our own data with oral fluconazole therapy in patients with ulcerative colitis who have detectable candida in the stool, a double-blind randomized controlled trial. And we had 242 patients of ulcerative colitis. We looked at the level of 28% were culture positive. They were randomized to fluconazole versus placebo for 21 days. And we looked at the various parameters and we found that active inflammation and steroid exposure increased the chances of finding candida culture positivity in the stool. And we found that when we gave fluconazole, there was a reduction in the disease activity in both the groups, but far more in the drug group than the placebo group. And there was a 3-point fall in Mayo Clinic. Mayo score was more often in the drug group. And we found that the calprotectin levels, which is a marker of gut inflammation, came down from 375 to 87, whereas in the placebo group did not change very much. And the histological improvement was also far more in the fluconazole group than the placebo group. This is the histology, representative histology, pre-treatment and post-treatment, resulting in significant reduction in the inflammation at the level of the mucosa. And the subjective feeling was also better in the drug group compared to the placebo group. So the key conclusion of this study was that in patients with ulcerative colitis, there was increased evidence of mucosa-associated candida, and candida positivity was over 28% and was associated with increased disease activity, and fluconazole therapy reduced the disease activity. So the last study, which I'll just quote here, is an inflammation-related differences in the mucosa-associated microbiota and the intestinal barrier function in colonic Crohn's disease. This study I did as a part of my fellowship at McMaster University at Canada. And here we started with the premise that patients who are having inflammation, so from the site of inflammation, we took a biopsy, and 5 centimeters away from a non-inflamed area in the same patient, we took a paired biopsy. And here, and we also had control population, and so that added as a control for our patients. And what did we find? That patients with this is the red one represents the inflamed area. The red one, inflamed area, had a different profile, microbiota profile compared to the controls. And those who had this is the inflamed area versus the non-inflamed area had a different microbiological profile. The inflamed area versus the control also had a different microbiological profile, suggesting focal dysbiosis in these sites. And we also found that the Chow diversity index was higher in the non-inflamed areas. And we found that the ones who had inflammation had very less diversity and more often pathobionts there. And this is very interesting. Can I just have 1 minute here? So this slide is interesting. And here you can see the colonic epithelium in those without inflammation. You can see the distance between the bacteria. This is fish. Compared to the epithelium, there's a wide distance because of the normal mucous lining. But if you see patients with injury, it is just the bacteria sitting on the epithelium. So here the bacteria is driving the inflammation because it has come close to the epithelial layer. And these are representative slides. Also showing this is a confocal microscopy showing that in IBD, there's a lot of inflammation and increased permeability and leakage of contrast, which we can see here. And more often, increased permeability at the tissue level can be seen. So the other studies which are ongoing or in patients with gallstones and seeing what is happening in the duodenum, and those in which we have put in a CBD stent occlusion with duodenal flora. And right now we are doing a study with Dr. Neelam. Arpita is the PhD scholar on organoid models. And we have also done studies on gallbladder mucosa-associated microbiome and the pre-neoplastic lesions in another study which my student has completed. So to conclude, Chairman, the mucosa-associated microbiome seems to be a key driver for mucosal inflammation. The sites for mucosal inflammation have a different bacterial profile, and there may be a role of focal dysbiosis needs further exploration. In diabetics, duodenal dysbiosis is common, and we should be targeting that. In patients with IBD, going beyond microbiome to look at microbiome and the role of candida may be useful. Antifungals may add another drug in the therapeutic armamentarium of this difficult disease. And understanding mucosa-associated microbiome may further our horizon in understanding gut microbiome and host interactions. Thank you. And this is my team at PGI, which has helped me with this work. Thank you.
Thank you, Dr. Usha. I think in interest of time, you can hold your questions till the end of the session. And now I invite the next speaker, Professor Ashish Sharma. The title of his talk will be Transcriptomic Studies on Donor Immune Cells to Reveal Genomic Markers of Kidney Injury. Dr. Ashish, please.
So thank you very much. At the outset, I would like to thank the organizers and Dr. Neelam Taneja for giving me an overview of what is happening in the microbiome area related to the gut. So I'm a kidney person, and I think maybe the society names now needs to change, and then you need to remove the gut because the microbiome is associated with all the specialties. And sitting here, I mean, this has not been my primary area of work, but sitting here, I could see that there is so much of need, even in our population where we deal with so much of antimicrobial resistance, especially in transplant patients, as well as the drug side effects. Like we see almost 30% of our population will develop GI side effects, which we attribute to the drugs, but which may just possibly be alteration in the microbiomes. So I'm going to take you a little different from gut to the kidney and from microbiota to microbiome. So please bear with me if you are not going to like it. So we do kidney transplants for patients who failed their kidneys. And so we were trying to develop a model because it's of interest to us that we have a huge shortage of kidneys. And not all kidneys, as we age, as we know that the microbiome changes, the kidney also changes. So as we age, there is a differential aging of kidney across the population. Some kidneys age much faster, some kidneys age slow. And there are injuries happening when we are taking the kidney out. We put the kidney either on the ice or on some machines, and then we could put the kidney back. So we thought it's a very interesting model of acute kidney injury. And that is what we try to analyze that. Can we get some markers which have not been reported so far or which have not been studied so far? Because this would be an ideal situation where you can actually develop this model of kidney injury and what are the genetic expression associated with different cells and what do you get out of the fluid. So we know that this after any organ retrieval, we take the kidney out of the body. So it's going to suffer a period of acute ischemia, hypoxia, which leads to a generation of free radicals when you perfuse the organs again. And at that time, all the immune system gets activated. You've got your antigen presentation from the dendritic cell in the kidney, and they express different genes. And that is what we try to assess, that we will just perfuse the organ, and when we get that fluid out, and we'll try to analyze what cell count or what expression of genes we are getting. So at the outset, we were very wary whether we'll be able to get any cell out of that fluid or not, because it is not blood. Now we are only perfusing kidney with the fluid. But we took almost 100 ml of that fluid. We split it into 3 parts, and one part we assessed for the cellular function, and the other 2 we assessed for the adherent cells after co-culturing, and the third part was just for the free cells which were not adherent to the culture medias. So we assessed the viability of these cells, and we found that if you are able to do this analysis within the first 2.5 hours, you get a good yield, but beyond 4 hours, the cell viability starts to decline. So what we did is that we tried to do everything within the first couple of hours. So these are results that we could see that almost 47% were the lineage positive and 50% were lineage negative cells. And out of the lineage negative, about 3.8%, about 4% were the dendritic cells, which we were interested because they are the cells which actually lead to all the immunological cascade in the donor, in the recipient, sorry. So this is our population. So 25 patients, the standard mean age of our donors. And we could isolate cells with more than 95% viability because we're doing it very early. Immediately after perfusion, we could actually process the RNA samples from these fluids. And these are the microscopic pictures, the sorted dendritic cells, the complete kidney cells, and the longitudinal ones are on the rightmost are the adherent epithelial cells. And this is the summary of the raw data. So so you can see that most of the reads were actually known ones, the mapped reads for all three populations, about 99%, but there were some unmapped and about some were uniquely mapped. So about 80-90% in the kidney cells and about 60% in the donor dendritic cells were the unmapped reads. So this is the principal component analysis where we could differentiate all the three cells, the adherent cells and the free cells and the dendritic cells. And this is the gene profile amongst the dendritic cells versus the adherent culture cells. So there's a huge expression, a huge number of genes which were differentially expressed across these samples. And these were actually involved in various functions, depending upon oxygen transport, gas transport, healing, repair, so many things. So upon enriching, we could find that the differentially expressed genes were mentioned in the bottom. And out of these, the TRIM58 and the KLF1 genes were the ones which were most important. And when we analyze these with the networking, we could see that these genes do not work in isolation, and they are associated with a lot of other activities and a lot of other proteins, which you can see in the network. And similarly, for the adherent epithelium cells, the number of genes were fewer. But out of these, we could find that the CD248 and the Lumican expression were the most significant. So these are the variables by the plot. Say you can see that there's a lot of significant difference in the dendritic cells in terms of KLF1 and the TRIM58, and these are the genes which are actually protective genes. That they are the KLF1 is actually reduces the inflammation which is going to happen. So my hypothesis is that when we are removing the kidney, not only the markers but the protective markers will also increase. So that is why these were upregulated. Same is for TRIM58, which is again an anti-inflammatory gene expressed by the dendritic cells. And along with these protective genes, there were the markers of injury, the Lumican and the CD248 were also upregulated. So because of paucity of time, I am not going to end the entire genetic analysis. So to summarize it, that this is the first analysis coming across the world where we've actually done this perfusion studies describing the mRNA analysis, and which we could find that there are injury markers which are expressed differentially across different population of cells. And these, I believe, that can be used to analyze the quality of these kidneys and which can help in allocation of these organs. But definitely we need further follow-up. It's just a work in progress, and we need some follow-up to see that how these kidneys behave after the differential expression of these biomarkers. Thank you very much. And this is our team, and the picture on the left is the ex-PhD person who's actually done all the hard work. Thank you very much.
Thank you, Dr. Ashish, for bringing transcriptomics into the conference. You have saved quite a lot of our time. But since I already announced that the questions and answers will be at the end, so we'll call you again. Thank you.
With this, I would like to invite Dr. Ashish Kakkad, who would be speaking on modulation of gut microbiome in neurological disorders, the pharmacomicrobiomics and beyond. Dr. Ashish Kakkad, please.
Thank you, ma'am. So at the outset, I'd like to thank Professor Neelam Taneja and her team for giving me this opportunity, and also congratulate her for hosting this wonderful platform focused on the research and clinical applications of probiotics. So I'll dive right into the presentation. So I'll be briefly talking about the actual problem that we are trying to address here with our studies, briefly focusing on the discipline of pharmacomicrobiomics, and then sharing my experience, and finally wrapping it up. So basically, when we start drug therapy in any patient, so these can be any of the four scenarios that can be met with. So of course, the one that we want is the one on the bottom, where we want our patients to benefit from those medications with little or no adverse effects. And on the diametrically opposite side is the zero benefit risk, where there are no benefits and no adverse effects, right? But more often than not, the situation is the one on the left or the right. That is, either there are no benefits or the benefits are being outweighed by the adverse effects, right? And what data shows us is that the approximate response rate for most medications that we use ranges from 50 to 75%. So about 1/4 to 1/2 of patients will not respond to the usual therapy or to the usual doses of drugs. And the cost of medicine-related harm has been estimated to be in billions of dollars. And the cost of ineffective therapy is yet to be estimated, but it's likely to be substantial. So if the diagnosis is correct and the patient is compliant to the medicines, then the most likely reason of this individual variability in drug response is because of several factors. And these are the various factors that we are known to us. Of course, genetics are the one which will come first to our mind. Of course, there can be variations in the drug metabolizing enzymes and transporters and targets. There can be drug interactions, factors such as gender and age, which is non-modifiable. And then we'll have disease states and environmental factors in one. But the one which we'll be focusing on today is, of course, the one on the right, that is gut microbiome or the gut microbiota. And pharmacomicrobiomics, as a discipline, if I have to describe it in one line, is how bugs, they modulate the drugs. It's basically describing the interaction between the gut microbiota and the drug response, which includes both its pharmacokinetics, that is absorption, distribution, metabolism, and excretion, and the drug pharmacodynamics, including the metabolic and the immune pathways, which can affect an organism's or an individual's susceptibility to the drug action. And besides, we all know a number of drugs can themselves affect gut microbiome, other than the antibiotics, which we all know can cause gut dysbiosis. But this study, which was published a few years ago, it found as many as 19 drugs, which were responsible for approximately 10% variability between the microbiome of individuals. So this figure summarizes in a nutshell the discipline of pharmacomicrobiomics as of date. And there are a number of conditions where drugs within the gut are modulated by the microbes, which affect the drug action. But today I'll be focusing on our research area, that is mainly in the central nervous system and nervous system disorders. So coming to our studies, so first is the study where we evaluated the impact of eradication of Helicobacter pylori on motor symptoms and quality of life of patients with Parkinson's disease. So Parkinson's disease, as many of you would know, is actually a neurodegenerative disorder involving destruction of dopaminergic neurons in the substantia nigra of midbrain. And the gold standard drug for that condition is, of course, levodopa, which is known to us for more than 5 decades now. And still it continues to be the #1 drug, which has been shown to be most effective in these patients. And what happens with levodopa is that a large percentage of levodopa gets metabolized in the periphery, and a very small percentage of it is able to reach the brain, approximately 1 to 3%. And to tackle that, we tend to inhibit its peripheral metabolism by combining it with a peripheral decarboxylase inhibitor known as carbidopa, which will still be able to enhance it to only 10%. So that is what is known about levodopa as on date. And coming on to the link between Helicobacter pylori and Parkinson's disease, there are a number of studies which underlie this hypothesis that H. pylori is linked to Parkinson's disease. So of course, there are epidemiological studies which show that there are high rates of H. pylori infections in patients with PD, ranging from 32 to 70%. And a meta-analysis of more than 30,000 patients showed that the H. pylori raises the risk of Parkinson's disease by 1.5 to 2 times. Of course, there are studies which have shown that patients with PD who are infected with H. pylori, they tend to have severe symptoms, which improve once H. pylori eradication is achieved. And this is the probable mechanism through which H. pylori eradication works. In the interest of time, I think we can skip this slide. And so before we did this study, we actually did a comprehensive literature review. And we found that while the observational studies here show clear benefits of H. pylori eradication in these patients, they could not be replicated in randomized controlled trials. And most of these trials, they did not show much improvement in motor or non-motor symptoms. And they could not replicate the positive findings of observational studies. And of course, there can be a number of reasons, as many of you would be aware of, why there are differences between the results seen from observational studies and why we consider the randomized controlled trials as the gold standard for evidence, right? So these are some of the reasons why. So there is so much variability in these studies. Of course, there is a substantial placebo effect, which is known to be seen in patients with Parkinson's disease. Of course, these studies, they varied in terms of H. pylori detection and eradication protocols. And finally, there was a possibility that this eradication, it may not be a true disease modifier. So this was the study that we planned. So we included both Parkinson's disease patients as well as healthy controls. And these patients and the controls, they underwent the urea breath test looking for H. pylori infection. And among both these groups, we separated these groups into H. pylori positive and H. pylori negative. Amongst PD patients, the H. pylori positives, we compared the symptoms between those who were H. pylori positive and H. pylori negative, and those that were positive then entered into a randomized controlled trial where H. pylori eradication therapy was compared with placebo to look for improvement in their motor and non-motor symptoms of Parkinson's disease. What we did found was that there was a significantly increased prevalence of H. pylori infections in patients with Parkinson's disease. That was 46% versus 26%. However, among patients who were diagnosed with Parkinson's disease, those who were H. pylori positive and negative, there was not much difference in their motor or non-motor symptoms as well as their quality of life. And also the levodopa dose between the two groups, HP positive and HP negative, was not substantially different. The results of our randomized controlled trials, they are currently being formulated into a manuscript, but this is a similar study which was published about a couple of years ago by Tan et al. And they also found similar results that over a period of 52 weeks, these results were there were no significant differences among the groups that were randomized to placebo or that were randomized to H. pylori eradication therapy. So be it the motor symptoms, non-motor symptoms, or the quality of life. So perhaps what it is hypothesized is that while H. pylori eradication, it may improve levodopa absorption, but those benefits, they do not, or they are not sustained over the period of, say, 52 weeks beyond H. pylori eradication. There may be earlier benefits, but later on, they are not sustained. Coming to another study that we are currently conducting is, we are evaluating the role of gut microbiota in modifying responsiveness to pharmacotherapy in PD patients. So already talked about H. pylori eradication. So we see that even when we inhibit its peripheral metabolism, only about 10% of which is able to reach the brain. And this is actually the reason why this is still substantial utilization of levodopa in the periphery, is that because of a couple of studies which were published in 2020. There were two different groups, one in US, one in Netherlands. And what they found was this levodopa is also broken down in the gut. So there is this enzyme tyrosine decarboxylase, which is produced by E. faecalis, which breaks down levodopa in the gut, reducing its bioavailability. And they gave this hypothesis that initially when we give levodopa, carbidopa, the bioavailability is high because there is a very small abundance for tyrosine decarboxylase, but with prolonged levodopa treatment and with disease progression and reduction in gut motility, there is increased colonization and increased production of tyrosine decarboxylase, leading to decreased levodopa bioavailability. Therefore, we planned this study to determine the association of gut microbiota composition and tyrosine decarboxylase activity with levodopa responsiveness in Parkinson's disease patients, and thereafter to target inhibition of this enzyme as a therapeutic strategy in these patients. So this is the basic study plan, where we will be enrolling both Parkinson's disease patients, responders as well as non-responders. We'll be measuring plasma levodopa levels at the peak of levodopa action, and also fecal samples are being collected, which are being subjected to gut microbiota composition and also tyrosine gene abundance by means of quantitative PCR. And what we have hypothesized is that there is going to be a negative correlation between levodopa levels in the serum of PD patients and the abundance of tyrosine gene. So higher is the levodopa responsiveness, it will be corresponding to higher plasma levodopa levels, and that will be inversely proportional to the tyrosine decarboxylase gene abundance. The next step in this study would be, if this works, is to develop an inhibitor of this tyrosine decarboxylase enzyme so as to improve the bioavailability of levodopa in patients and to improve their clinical outcomes. So one of the initial work that we did in this study was we wanted to hypothesize, wanted to see if carbidopa, that is a peripheral decarboxylase inhibitors in humans, can it also inhibit tyrosine decarboxylase in the gut. And we did this an in silico study, and we found that the binding affinity of carbidopa for the tyrosine decarboxylase is quite low, and it perhaps cannot inhibit this. And therefore, we have to have another inhibitor which can specifically target tyrosine decarboxylase so as to improve levodopa bioavailability. Finally, the third study is modulation of gut microbiome in drug-resistant epilepsy. And we know that nearly only two-thirds of patients with epilepsy will respond to the first-line therapy, and nearly one-third of the patients, they will be classified as drug-resistant epilepsy. And there are a number of reasons how gut dysbiosis contributes to the pathogenesis of epilepsy and also the drug-resistant epilepsy. So it is related to the release of neurotransmitters, synthesis of short-chain fatty acids, as well as interference. I'll just take a couple of minutes. Endocannabinoid system and vagus nerve signaling. So in the interest of time, I'll just go right to our study. We hypothesize here that intervention with high-potency multi-strain probiotics will modify the gut microbiome in patients with drug-resistant epilepsy, leading to improved seizure control. So this is basically our study where we'll be randomizing these patients to the probiotic intervention group and to the placebo group. They'll be followed every 4 weeks for 16 weeks. And we'll be looking at the clinical symptomatology as well as gut microbiome, as well as the blood levels. So we'll have an intervention phase of 16 weeks and a post-intervention phase of 12 weeks, where we'll look for sustained effects of these probiotics. So we originally wanted to do this study with the De Simone formulation, and we reached out to Professor Claudio De Simone, who himself suggested that a more potent formulation that he has created, that is Sevomix, which is nearly twice as potent as the De Simone formulation. And we are currently conducting this study with this formulation, which is a double-blind placebo-controlled trial. Of course, the primary outcome is clinical, where there is a percentage reduction in seizure frequency. And these are the other secondary outcomes for our study. So far, we have been able to recruit two-thirds of our patients with a mean age of 30 years, and nearly 50% distribution for both genders. The mean seizure rate in these patients is 6 per month, and they are taking on an average of 3 to 4 other epileptic drugs. These are the most common anti-epileptic drugs they are receiving, and at the bottom you can see the most common adverse effects being experienced by these patients because of these anti-epileptic drugs which they are taking. So preliminary analysis has shown that the most common bacterium phyla have been the firmicutes and the bacterodata, which is in line with what we have seen in literature, that there is increased abundance of firmicutes in these patients with epilepsy, especially with drug-resistant epilepsy. And therefore, we also did a preliminary analysis of the benefit of probiotic supplementation in these patients, and we found that there was 10% reduction in seizures in these patients. And importantly, 25% of these patients with epilepsy who received probiotic intervention, they had more than 50% response rate, which means that their baseline seizure frequency came down by 50%. So these are some of the challenges in fibro. This is going to be a last slide. So of course, many of these factors have already been discussed since morning. So I'll not be diving into them. And these are some of the ways to boost pharmacomicrobiomics. So these are my acknowledgements. So on the right are the colleagues in my department and the students who have been working on this. And the yellow are my collaborators from other departments, from medical microbiology, neurology, biophysics, and gastroenterology. And on the extreme right-hand side are the funders who we acknowledge for the support. Thank you very much.
Thank you, Ashish. I know you've done a lot of work and there was time constraint. So just can you sit in this front row?
Yeah,
The
sure.
questions and answers.
I'll be there.
The end. So now I invite Dr. Amit Arora, and his topic is relevation of the breast milk microbiome of Indian mothers. Amit is a bioinformatician and a faculty of our department. Amit, please.
Thank you, ma'am. First of all, thanks to Professor Neelam Taneja and the organizers for this invite. So yesterday, Professor Saurabh Datta gave a wonderful talk about the milk microbiome and it was pretty revealing. And I've been working with him primarily for this work. And this is a multi-year work and a couple of students are also involved in this. So let me just... So everybody talks about the gut. So gut is, of course, the reservoir. Gut is the storehouse where all the microbiota resides, and many factors affect the gut. We all know, we have all discussed this since many years. Everybody knows drugs affect the gut microbiome. The food that we take, the exercises, the age, everything affects the gut microbiome. But nowadays, what is happening is we are coming to know that there is an axis. There's an interconnection, there's a crosstalk between the gut and different organs. And a lot of publications are also coming that the gut microbiome actually affects a lot of other organs and their functionality and their well-being. Recently, it has, like Professor Saurabh Datta also showed, that there is strong evidence evidence of the mother's gut and the mammary gland interactions are also there. So this has not been studied very well. One of the papers is this Perez et al., in which what they showed was that the bacterial signatures in the feces, milk, and the blood of the mother are identical to the feces, to this bacterial signatures in the feces of the infant. So how did that happen? There has to be a connection. There has to be some kind of mode of transmission. What do we know? There is very little information as far as the milk microbiome is concerned. People have started working, but the kind of information that is required is still lacking. We know that the milk production starts after 16 weeks of gestation. We know that the premature breast milk microbiome differs to the term mother's milk microbiome. There is a distinct difference. We know antibiotics affect everything. Of course, antibiotics, by definition, they are here to kill the bacteria and they affect the microbiome. Diet is a very big association. We know that different regions in the world, they have different food and the microbiota is different. There's a Western diet and there's a non-Western diet in which the microbiota is known to be very different. Also, we know that it is known that the obesity, especially in the Western population, obesity is a major factor. But in Indian population, low BMI and underweight mothers is also a very major problem. So what we did was we took these 4 factors and we decided to investigate what more can we do, what can we actually explore in the population of Indian mothers. These are the numbers that we took. This is just a slide just to show the numbers. So we had a gestation term, more than 37 weeks is term gestation. 26 to 28 is extremely preterm, and 29 to 31 weeks is very preterm. These are the BMI values. This is the diet, vegetarian versus non-vegetarian. Like, in India, we do get, equal kind of proportion of vegetarian to non-vegetarian people. Therefore, this is what we took. And antibiotics, these are the numbers that we have. Now, some people say that these might seem slightly less. But the point is here, if we are collecting milk from preterm mothers, for preterm mothers, the milk development hardly starts. So the amount of milk, the quantity of milk that we get is very, very less. So the student, I think Anvesha might be sitting over here somewhere. She collected milk from preterm mothers and collecting that milk, sitting whole day, and whatever drops came out from the mammary glands, that was collected and that was sent for analysis. So sample size, the quantity, the quality of sample that was collected is very, very difficult. Therefore, this study took a lot of years. And that was one of the main reasons that it was a very difficult study. What we did, everybody's talking about 16S metagenomics. Everybody knows about that very well. Professor Ganguly also just mentioned there was a talk where, there was a difference between 16S abundances and WGS. we did whole shotgun metagenomic sequencing. What happens is after collecting the milk, we isolated the DNA and we gave it for sequencing for a whole shotgun metagenomics. Now, what do I mean by whole shotgun metagenomics? 16S, basically what we are targeting is we are targeting the V3, V5 region of the 16S rRNA gene. And that is just a small proportion. What do we get out of that is what is there and how much of it is there. Beyond that, we don't get any other information. We know we will, from this approach, we'll only get what is there and how much of it is there. But from shotgun metagenomics, what we are sequencing is everything, the whole DNA. We are sequencing not only the 16S, but the rest of the genes, the rest of the DNA, whatever is there, we are sequencing everything. So not only do we get the abundance levels of what is there, we also get better species level resolution because it is just not one gene. It is a set of markers that those are used actually for your annotation and taxonomic annotations and diversity analysis we get. And we also get functional information, something like pathways, which I'm going to show just now. Before I go on to present the results, I'm going a bit fast just to conserve the time. Something about diversity. What is diverse? So diverse populations of microbiome are good. The more diverse the microbiota is, the more resilient, the more robust the organs are, the more robust the host is to fight off diseases and to be, generally strong. So this is just something which I wanted to show, but I'll just go forward. I'm sure people know about it. So what are one of the first things, although this is not statistically significant, but we found out that the term mothers had higher diversity compared to the preterm groups. Right? So this is, it was expected, but, we were just exploring initially, how does it work out? BMI, the higher BMI, the mothers with higher BMI, the mothers having a higher BMI had higher diversity. Again, very, very interesting. This is kind of a proof that, yeah, higher BMI and term gestation period is actually beneficial even for the milk microbiome. Diet, non-vegetarian diet. Now, again, there's a lot of discussion in, vegetarianism versus non-vegetarianism, but this is something which is very interesting. How does a diet actually affect, and I'm going to show something very interesting later on also, how does the diet affect the microbiome? We found out, and this was significant, we found out that the mothers having non-veg diet during pregnancy had higher diversity. These are just few of the results. We have many other things. Just to conserve time, I'm just showing one of the main results over here. Again, fourth thing is antibiotic exposure. As expected, the mothers which are not having antibiotic exposure had higher diversity in their breast milk microbiome compared to the ones having 1 to 3 days or more than 5 days antibiotics. As I told you, what we are doing shotgun metagenomics. The beauty is we can map the genes that we got after a lot of analysis. We can map the genes and map them onto the functional pathways. How does the body function? Body functions, they are individual genes, but individual genes interact with each other and form functional pathways. And what we found out was this was done for all the pathways, all the metaboloids, thousands of pathways, literally millions of genes. And this is, what we found out is as far as gestation age is concerned, the valine biosynthesis pathway was significantly expressed. The genes in the bacteria, in the milk, were significantly overrepresented in the preterm groups compared to the ones that are there in the preterm groups. And it is already known that valine concentration was found to be higher in preterm breast milk, in the preterm breast milk. And these are all statistically significant and multiply tested adjusted Q values. So this is quite significant, not just at the P value, but also at the adjusted P values. Again, from the previous result, we know that preterm mother's milk has more protein content. We know that several amino acids, valine, for example, is found more in the preterm milk. Earlier, it was thought it is just a leaky gap junction that actually, permits proteins to come into the bacteria. But no, is there a microbiome association also with it? Because we found some very good, interesting results over here. Secondly, we found, pantothenic acid with vitamin B3 biosynthesis pathways to be extremely overrepresented again in the preterm group versus the term group. Again, I will show this with an evidence, what is known previously, that pantothenic levels are in the preterm milk are significantly higher. Again, the question is it microbial in origin? Coenzyme I, after pantothenic acid, we have the coenzyme IA, which is synthesized from vitamin B5 from pantothenic acid. So again, the same story is there, again, very significant. What we know that atopic dermatitis is associated with higher coenzyme IA content in the milk. But, I talked to my dermatologist over here, I talked to a couple of people, although there's an inverse correlation between preterm birth and atopic dermatitis. If it was a straight correlation, I would be very, happy about it, but it's an inverse correlation. But still, this result is also pretty interesting. Another thing is that, bacteria use acetate and lactate and get basically energy. So accumulation of lactate in gut is associated with IBD and UC in infants. And in preterm infants, the lactate and acetate levels, the bacteria having these pathways were significantly overrepresented. I think Professor Usha Datta could really elaborate on this. This is what we found over here. Whether it is significant as far as preterm births are concerned, that can be discussed. Diet. I think Professor Ganguly, asked the question yesterday that, what is the significance of diet as far as the composition of milk is concerned? There's also speculation that the Indian milk somehow is not of that good quality as the Western Caucasian, milk in its composition, not only as of microbiota, but also the protein content and nutrient content. We found several pathways. Now, this was not significant at the multiple testing level. Of course, if we have a bigger sample size, this could be. We found that the pathway for lactose and galactose degradation was significantly higher in veg mothers. Now, veg mothers, I feel, would be having a lot of milk products and milk. So this makes a lot of sense over there also. Again, an association with the microbiome and the genes that are there in the microbiota that are actually being functional. Another thing which was very interesting which was actually pretty shocking to me, that, this is Calvin-Benson-Basham cycle, which is a plant-based cycle, which is, CO2 fixation pathway found in all green plants. And this was again significantly overexpressed. The genes in this cycle were significantly overexpressed in the veg mothers, not at the multiple testing level, but definitely at this level. More sample size could give us some more information. Yesterday, this slide I took from Professor Datta's presentation, and he showed the difference between, I think it was a study from Gujarat, then there was a Finnish study and a Switzerland study, and there was also a Spanish study, which he showed yesterday, in which there is a marked difference between the microbiome of mothers from India versus the Caucasian population. So yes, there are differences, but because I found out something with the diet, region, all the regions, even in India, every 200 kilometers, there is a different diet. Could it be related to diet? These things, I'm sure there is a very strong correlation in this. This is again from yesterday, but I'll just show you something, antibiotic exposure, because this is a very important topic. So what we had over here was, we have mothers without having any antibiotics. We have mothers, preterm mothers without antibiotics. I think this was 1 dose of antibiotic, probably this is a typo. This is 1 to 3 days of antibiotics, and this is 5 days of antibiotics, more than 5 days of antibiotics. What we have over here is, let me explain how to interpret this diagram. The height of the blocks corresponds to the hits. Now in NGS, we get reads. Reads are small and the gene is big. An antibiotic resistant gene is big, but the reads are very small. So these are basically, the height represents the number of reads that are actually getting hit, that are getting mapped onto that gene. And the color represents how much of that horizontal gene sequence is getting mapped. So genes could be 1,000 base pair. Is it, are we getting 500 base pair, 800 base pair, or all of 1,000 base pair? So this is what it represents, 2 things. But again, the take home message is, look at the difference in the breast milk, in the genes of the mothers having antibiotic This is just a summary of the points that I've showed you. Again, Professor Saurabh Dutta, excellent questions, excellent collaboration. I don't think he's here right now, but a wonderful collaboration with him and Vesha. She did all the hard work, sat from morning till night, collected the milk from preterm babies, from preterm mothers, very, very difficult. And of course, the neonates and the mothers who really donated the milk to us so that we are able to do this study. And we took milk from them. Thanks to them a lot. Thank you very much.
Thank you,
Amit.
Dr.
Can
Amit,
you-
for the fascinating journey on milk microbiome. We will have the questions after this session, and it's my pleasure to now invite Dr. Nipun Verma, who's an associate professor in the Department of Hepatology at PGI Chandigarh. Dr. Nipun has been actively working on fecal microbiota transplantation in patients with ACLF, as well as his work on fungal infection in patients with liver disease. Over to Dr. Nipun to speak on harnessing circulating omics, a new frontier in tackling infections in cirrhotics. Dr. Nipun, please.
Thank you very much, ma'am. At the outset, let me thank Professor Neelam, the esteemed chairperson, Professor Datta, esteemed audience, for being here on Sunday morning for the lectures. My topic today is circulating omics in patients with cirrhosis, and I bring greetings from Department of Hepatology, PGI. So why I'm interested in infections in cirrhosis, I'll come to that, but briefly describing the overall points of discussion of my talk would be that why infections are important in patients with cirrhosis, what is the epidemiology and impact of infections, how we can harness the power of omics, especially the proteomics and metaproteomics, and what lies in the future. So we understand cirrhosis and liver. Liver is an important organ in human body, actually taking into not only a metabolic organ, it actually takes into account different pathogens, its products into the circulation, and it prevents from gut translocation, causing the systemic immune dysfunction. So these patients with liver cirrhosis have extensive fibrosis, cell death, Kupffer cell dysfunction, dysfunction in the synthetic proteins such as complement, acute phase, reactants, causing lot of gut pathogens to transmigrate into systemic circulation. The Kupffer cell dysfunction incites lot of inflammation inside the liver, which actually spills over to systemic circulation, heightens the susceptibility of different organs to pathogen-related damage, immunopathological damage, and multi-organ failures. So in patients with cirrhosis, it is a unique condition. In this, patients compared to without cirrhosis have higher risk of infection, about 4 to 5-fold. And these patients are also at higher risk of multi-drug resistant infections. These patients at higher risk of end organ damage, leading to multi-organ failures. And we call this syndrome as acute on chronic liver failure. And often the delays in diagnosis results in high mortality. Fungal infections are rising and leads to high and exorbitant mortality in these patients. And we know that over the years, the mortality related to infections is not decreasing. So it is very characteristic of these patients with liver cirrhosis. When they die, their natural history suggests there is a phase called acute decompensation, where these patients developed severe onset of ascites, encephalopathy, varicose bleeding, and infections. Some of these patients, about 30% of them, progress on to organ failures, and we call these patients as acute on chronic liver failure. These patients, the mortality ranges from 15% to 100%, with an average mortality of 50%. So in these group of patients, the infections are quite common. These infections, as of the largest data available till date, suggest that 70% of these infections are multi-drug resistant. And it is concerning that carbapenem resistance has increased over the last few years to the 50%. The important risk factor for infections in these patients include the colonization, as we heard yesterday, Dr. Vineet, and we have also understood that these patients with cirrhosis at admission are 70% of the times colonized with multi-drug resistant bacteria. And these multi-drug resistant bacterial colonization is linked with 4 times higher risk of developing multi-drug resistant infections. So why these patients are at predisposition to multi-drug resistant infections is a question in concern. And we also know that patients who have infections, whether it be multi-drug resistant or presence of ACLF, the mortality is much higher. So apart from several factors which govern the risk of infections in cirrhosis, the most important factor, as we have heard over the last one day, is microbial factors. So gut dysbiosis, lot of literature exists. Extensive dysbiosis, cirrhosis dysbiosis ratio, a lot of studies have been demonstrating that it is linked to the severity of cirrhosis, the severity of decompensations, patients who are progressing on to mortality, all such factors, and extensively studied the gut dysbiosis in patients with cirrhosis. There has been a lot of literature on gut barrier failure, lot of literature including the mucosal permeability, mucosa-associated lymphoid tissue, GALT, a lot of studies. But there are limited studies as to interactions between this gut dysbiosis and what is happening inside the circulation. So cirrhosis-associated immune dysfunction, as I said, is because of cell death, extensive cell death inside the liver. And this leads to Kupffer cell damage as well, dysfunction. And this dysfunction is continuous. So patients who develop cirrhosis, they develop it over the years. So there is a constant flux of microbes, not only bacteria, viruses, and fungi, constant stimulation of Kupffer cells and leading to the immune paralysis in Kupffer cells. This immune paralysis incites secondary infections, more so fungal infections and drug-resistant infections. So lot of literature needs to be focused on what is happening inside the circulation and how it is impacting the patient outcomes. Apart from that, there are several clinical factors which govern the risk of infections, which include the severity of cirrhosis, the severity of liver damage. So our question in this scenario, in patients with liver cirrhosis, where there is a lot of systemic inflammation, a lot of cytokine storm, which is happening because of ongoing flux of pathogen-associated molecular patterns, there is a high difficulty to diagnose which patient is infected and which patient is not infected. This leads to overuse of antimicrobials in patients who are admitted in hospital. This overuse has been linked to multi-drug resistance. Moreover, patients who are not showing characteristic or typical signs or site of infection, these patients receive high-end antibiotics such as carbapenem. This also leads to the increased risk of multi-drug resistant infections. And our aim of the research was, can actually this circulating proteomics decipher which patient is infected in a highly inflammatory stage of infection and which patient is likely to develop infection later on. So our story started with proteomics to see the host response, and then we went on to microbial factors and metaproteome, which actually deals with the proteins, circulating proteins related to bacteria and fungi in patients with cirrhosis. So coming to the host response to the proteins, what we did in this study was we collected patients with acute decompensation and acute and chronic liver failure with multi-organ failures with and without infection. These patients with proven infections, and these were proven non-infected cases, we did untargeted high throughput LCMS through Orbitrap Fusion, which is a high-end machine. And we did this downstream analysis through several extensive bioinformatics statistical tests ranging from U-test, volcano plot, clustering, machine learning algorithms such as extreme gradient boost. And we found different host response factors which were distinguishing patients with and without infection. And then we jumped on to pathway analysis and how these proteins are involved in biology of the patients. After the discovery phase, we went on to recruit 3 different cohorts, 2 from PGI and 3rd from external center, and about 150 to 170 patients in each cohort. We validated these proteomic signatures as to which patients are infected and which patients not. And this is the result. We showed that about 27 proteins were upregulated and 38 were downregulated. And you can see that in the clustering analysis, the infected and non-infected patients are largely separable with minimal overlap. And we can see that in the principal component, the PLSDA factor loading plot, that a protein which was galectin-3 binding protein was significantly upregulated in patients who were having infection. And this was consistent in this extreme gradient boost, the machine learning algorithm as well. And this algorithm was 96% accurate in diagnosing infections. And in the validation cohort, what we showed that this protein was significantly up, and there was a gradient from healthy controls to the infected patients. And we also found three other proteins, which were different in patients with infections, but these proteins were downregulated, which were phospholipid transfer protein and the glutathione peroxidase. But the important point was that when we use different signatures, combine different signatures in statistical algorithms, we could find that a model which was a combination of a host response to infection, that is galectin-3 binding protein, and procalcitonin with liver organ failure score and lactate, performed very well in distinguishing these patients. And we could demonstrate that this was significantly superior in terms of distinguishing infection in patient with or without ACLF, with infection and without infection in ACLF. And this was superior as compared to procalcitonin and other clinical factors. Now, we also developed this calculator, which can be used at bedside to help our patient and prevent the overuse of antimicrobials. And these are the list of functions which these proteins do. Galectin-3 binding protein is something which is an extracellular glycoprotein. It helps in inciting the pro-inflammatory response, the neutrophilic phagocytosis, as well as the oxidative burst in neutrophils. And this protein, when increased in patients with infections, have shown to be protective in not only in patients with cirrhosis. The galectin-3 binding protein has been demonstrated in patients with without cirrhosis, with COVID, with fungal infections to be elevated. We went on further to describe the differences between patients who had SIRS and without infection, and we could show that there was significant shift in the metabolic usage of fatty acids as a source of energy. However, when we found that patients who develop infection followed by SIRS, their phagocytic pathway, the phagosome pathway, is overactivated. However, the patients who develop loss of systemic inflammatory response, there the phagocytosis pathway and the tight junction pathways were downregulated. And there is a host of differential factors which we could see that there were different between patients with and without infections. Further, when we compared bacterial infection from fungal infections, we could see that the two important biomarkers, sex hormone binding protein and SERPINA4, which is a cholestatin mediator, actually were differentially expressed in patients with fungal infection. And we also found the signature specific to multi-drug resistant organism infections like dermicidine and MASP1, which needs further validation. Now, we also know that fungal infections are increasingly recognized, and based on our several small studies, the descriptive studies, the systematic review also showed 15% of patients with cirrhosis have fungal infections. These patients have high mortality. We went on to study the circulating proteins related to Candida and how these proteins are linked with infections and outcomes of patients with cirrhosis. And what we found is an interesting protein called Candida leucine aminopeptidase. This protein, when combined with beta-D-glucan, showed a highly accurate marker for diagnosing infection up to 96%. And in subsequent studies, we have further validated this protein. I'll show that also. It was 93% accurate alone in per se diagnosing invasive candidiasis. Not so, but also in patients who are dying, these patients have higher levels of leucine aminopeptidase, and you can see in the survival curve, patients with high leucine aminopeptidase, the survival is just 7%. We also proposed, this is an hypothesis, that this leucine aminopeptidase acts at two levels, intracellular on fungus, involved in autophagy pathways, improving the survival or the fungal sustenance. Whereas in the extracellular pathways, this LAP2 actually acts as a peptidase, destroys the the prostaglandins, as well as increasing the pro-inflammatory response, and that is the hypothesis we have gave based on its functions. We also expanded this metaproteomic approach to through different statistical and machine learning algorithms in broader set of patients, about 100 patients, compared to healthy controls. And what we found that about several of fungi related proteins, especially this analysis was largely focused on the candida related proteins. We could see that there is a huge separation between patients who are surviving and those who are dying. So basically candida related proteins are actually making a difference between patients who are surviving and dying. And these different proteins were then mapped where they are located. They were located in cytosol, mitochondria, cell surface, and nucleus. And they were involved predominantly in the metabolic reprogramming of candida, morphogenesis and invasion, and transcription and translation of proteins inside the fungi. So this was the concept diagram we have made, which is showing that these proteins are involved in excessive glycolysis, the TCA cycle and the ETC chain, which promotes the fungal growth. These proteins are also involved, like GCN5 is involved in the the filamentation of fungi, and LKH1, which was highly upregulated, is involved in the invasion of fungi. And now we further moved on to see what are the microbial diversity changes at the level of Candida as a genus, and how it is different between patients who are surviving and non-surviving compared to healthy controls. And we
Dr.
could see.
Nipun, in the interest of time, you may have to summarize because we have.
Okay, ma'am. So this is the last two slides where I wish to describe that, this is the Candida albicans. As you can see there is a gradient that the Candida albicans related proteins are much higher in patients who were dying at the end of 30 days. And interestingly, you can see that Candida auris related proteins, even these patients were clinically not infected. Even then, these patients had these Candida related proteins circulating inside patients with ACLF. And here what we showed is that it's very interesting fact. We say that the diversity in the gut is important for a healthy gut. But here we found just the opposite. We found that the diversity at the level of alpha diversity, Shannon or Simpson index, was much higher in patients who were non-surviving. This implies that a lot of Candida and Candida related proteins are circulating inside the circulation in patients who are dying, who are sicker patients of ACLF. And on beta diversity, this can actually demonstrate that these proteins were segregating patients who are surviving and non-surviving on beta diversity plot. So we are now working on validation of such metaproteomics and proteins related to bacteria and as well as fungi. And we are going to develop different point of care devices to detect at bedside for diagnosis and prognosis in patients with ACLF. And we are going to look at multi-kingdom interactions, in vitro modeling, phage interaction, novel therapeutics. I would like to thank my team, Pratibha Garg and Rohit Nada, who are the students actively involved in this, and Dr. Neelam Taneja, who has been guiding, and Dr. Arna Lok and Dr. Shiva Prakash, who have been guiding us in this journey. Thank you so much.
Thank you, Dr. Nipun. With this, we will all agree that we have had 5 different disciplines of medicine all showcased here, from gastroenterology to nephrology to neurology to neonatology and hepatology. So this just shows the importance of gut microbiome and gut barrier in maintaining human health. And since there is lack of time, what we could do is we could have 2 questions per speaker from the floor for each of the speakers. And I would invite all the 4 speakers to come up so that we can have questions directed to them, or you could use the mic from below and speak from there.
Very full piece, and really impressed the work which is being done here. Neelamji, it was good that you set up this particular area. And I will make one brief comment and ask questions. Like Nipun's work was very nice. There is the similar work from ILBS. He showed Dr. Sarin's thing, and Jassi there does the proteomics. So here, Nipun, this galactin-3 binding protein is very diverse. Like we have published series of papers in glioma, and we have patented the system. So it has got a lot of other functions. So you could perhaps explore them. And Dr. Datta, your work was that is something which is amazing, that I saw in IBD patient tremendous amount of cryptoglybercune, which actually generate new epithelial cells. This was a miracle. I have never seen this kind of thing before. And finally, one last comment for Dr. Ashish Kakkar, that it will be useful to really look at the drug levels in epilepsy as well as in other, because they might be broken down. So apart from what we are seeing, the microbial population would perhaps prevent this drugs being broken down. He had made this hypothesis in Parkinson's disease. It could be the same in the epilepsy, but it was a tremendous thing. And Nipun, congratulations for publishing in Journal of Hepatology. This is one of the very high impact journals.
Thank you, sir. Yes.
My question is to Dr. Ashish Sharma. So the genetic cells you saw in these old kidneys, what was the phenotype of these genetic cells, and were these genetic cells able to activate the cells of recipients in autoreactive manner?
Only the first phase of the study, and then the second phase is going to do this, that the dendritic cells we are going to culture with the recipient cell and to see whether they are going to activate it or not. At the moment, we only saw the dendritic cell markers and their expression.
Second question is for Dr. Amit Arora. So, use this microbiota, which you saw in the milk, does it have any impact on the presence of IgG antibodies in the milk?
We have a lot of information regarding that. I did not present because there's a lot of information. There's a lot of clinical data that was also collected. I do not have the answer to your question right now, but we have that. We are compiling all the data sheet. But I guess maybe in a couple of days we can answer that. Not only that, we are also taking this project further. We have yesterday we got the QC results for RNA-seq. So this was the genomics work. This was metagenomics work. We are also doing metatranscriptomics, and we got some yesterday only in the evening we got some very interesting results for that. And it is continuing so that we actually know the functional part of it, whatever is going on. Right now it's all.
Thank you. Yes.
This question is to you, madam. Is that you showed that in the duodenal bacterial growth in diabetics. Now, if the glycemic control is made better, will the bacteria go away? Or if you treat the bacteria with antibiotic, will it improve the glycemic control?
Yes, very important question, which is the chicken and the egg. And that is the reason in India, we had an entity called tropical sprue, which was basically excess of bacteria in the small bowel compared to what we used to see in our Caucasian counterparts. So here we have tropical sprue has kind of disappeared because there's a lot of over-the-counter antibiotic use in India. And as a result, the bacterial load is not so much enough to cause significant disease, but maybe that has got translated to a lower bacterial load associated with the gut epithelium, especially in the duodenum, which is now manifesting as the diabetic capital of the world. So here I do think that the bacteria is driving the pathogenesis of duodenum. In fact, we have right now an ongoing study on looking at the eye cells, which is in the duodenum, and how this microbiota affects the eye cell signaling. It is after a diet, the eye cells trigger CCK release and secretin release and make the pancreas kick in. So if that crosstalk is not happening, we are going to have more of these endocrine problems related to diabetes. So I do think our study does not show what is the chicken and the egg, but the next phase of the study is to give antibiotics and see whether it helps in diabetic control. Yes, yes, madam.
Hello. My question is to Dr. Amit. As we know that gut has phyla, Bacteroidetes and Firmicutes. So in your study, Proteobacteria was the main phyla which was found. And Professor Saurabh also mentioned that only one study they found with the similar findings. And that too, Pseudomonas species was the major one. So is it like in two more cohorts, the phyla was again Bacteroidetes and Firmicutes, and only in your study?
The time I did not really go into the abundance levels and the taxonomic file. So that would have been
Dr. Amit, please keep your mic close to your
So I
mouth.
did not cover the abundance levels and the taxonomic profile that we got. So what Professor Datta told yesterday, we got similar profiles, but the things that you saw in the pathway abundance, measures, those were the pathways, those were the organisms in which those pathways are there. But that cannot be correlated to the abundance levels as such. The taxonomic part I have actually deleted from this presentation, but it was similar.
I have one question for you, Dr. Amit. Very interesting data. The non-vegetarian diet had higher diversity. Most of the people in India who are non-vegetarian, they are also consuming a fair bit of vegetarian diet. So maybe is it because of that they have higher diversity? They have the vegetarian and the non-vegetarian component?
We need to do a trial for this probably. So we don't know. That is very interesting because this was a component which was kind of added at the last minute. Let us see how the diet affects it. Because, from the gut to the mammary glands to the genes that are there in the microbiota, it's a very long shot. So what we got, there are a lot of pathways that are there. I'm just showing the significant ones. There are a ton of pathways there in each of these, antibiotics and the diet and the BMI and the gestation age. There are a lot of pathways. But these are just the top ones that I'm showing over here. So I do not know what is the answer to that.
Thank you. Yes, can we have any other questions on the floor? Yes, sir.
For sclerosis as well as Parkinson disease, what is the probiotic dosage actually is prescribed? Like for ASD, autism spectrum disorder, there is somewhere 200 billion count of the bacteria which is given, and then there is actually a good amount of recovery is much better. So such probiotic formulation is available for ASD. So in that one.
Dr. Ashish, do you want to answer that? The amount of probiotic which is required.
So I'm aware of what we are evaluating. So we are doing this for epilepsy with patients with drug-resistant epilepsy, and we're giving them Sevomix 800, and we're giving them one sachet twice a day, once in the morning and once in the afternoon. And I'm not sure if that dose has been standardized for the conditions which he was mentioning.
Okay. Do we have any other questions? If there are none, I think we should come to a conclusion of this session. And thank you all for active participation, and thank you to all the speakers who have done an excellent job.
Thank you. I may now request our chairpersons to felicitate the speakers. I request Professor Neelam Taneja to felicitate Professor Ashish Sharma and Dr. Ashish Kakkar. Can we have a huge round of applause? Let's have a huge round of applause. I request Professor Usha Datta to felicitate Dr. Amit Arora and Dr. Nipun Verma. Can we have a huge round of applause for both the speakers, Dr. Amit Arora and Dr. Nipun Verma? Let's have a huge round of applause. Thank you. May I request Professor Ganguly to felicitate our chairperson, Professor Neelam Taneja, and Dr. Neerja Hajela to felicitate Professor Usha Datta. Can we have a huge round of applause for our chairpersons? Okay. So I request Dr. Neerja Hajela to felicitate Professor Neelam Taneja and Professor Usha Datta. Let's have a huge round of applause. Let's have a huge round of applause. Let's have a huge round of applause once again. Thank you. I request you all to fill up the post-symposium survey form. Please have it filled and hand it over to the girls at the registration counter. And now we have a short 10 minutes break for tea and coffee. And I request you to please be back in the next 10 minutes because we have another interesting session coming up for you. Thank you.
Anil, check, check, 1, 2, check, check, 1, 2, 1, 2, check, 1, 2, check, check, check. ठीक है.
check number. Hello. इधर आइए. Check. Hello. मुझे 14 नंबर प्लस का है ना, प्लस का प्लस. हाँ, ये दबाइए नंबर. Check. Hello. Sound check. Mic check. इसको नीचे जाके ना check. Hello. फिर वो जाते विस्लिंग होते हैं ना. Sound check. Hello. ये हो गया. इसको check. इधर check. Sound check. 65 नंबर पर 65 का नीचे का.
Uh,
India Probiotic Symposium. So now, before we have the top 5 young investigators presentation, let us have a huge round of applause for all the young investigators who have submitted their abstracts. So can we have a huge round of applause for all of them? Thank you. So this was a very important part of our program. Out of 56 abstracts that we have received, the decision to selecting and finalizing top 20 was a difficult one. Such was the quality of the research work and content that we decided to invite the additional 15 more for displaying and showcasing their poster. After the morning poster evaluation session, the judges have shortlisted top 5 for oral presentation, and on the basis of their oral presentation, we will select our top 3. So for this, now let's have a huge round of applause for our top 5 investigators. Yes, can we have a huge round of applause for our top 5 investigators? So now for this, I would like to invite our chairperson, Professor Jyoti Tamang, Vice Chancellor Officiating, and Senior Professor Microbiology, Sikkim Central University, Gangtok, Sikkim, and Professor J.B. Prajapati, Chairman ITA, Mumbai and Coordinator, SASNET Fermented Foods, Anand, Gujarat, India, for taking over the session. So let's have a huge round of applause for Professor Jyoti Tamang and Professor J.B. Prajapati. So each investigator has 7 minutes as a timer. Thank you.
Welcome to this very, very important session. We have 5 brilliant scholars who have come out after a very serious screening. And as informed, we'll have 7 minutes of presentation. And after that, we can have 2-3 very, very relevant questions if there are. So I will first invite Mr. Rajdeep Dalal. He is from THSTI, Faridabad, and his work is on gut commensal derived succinate impels colonic inflammation in ulcerative colitis. Rajdeep. 7 minutes. Okay.
Yeah, good afternoon, everyone. So first of all, I would like to thank to the organizing committee, Professor N.K. Ganguly and Nirja ma'am for giving me this opportunity. And today I'm going to present my work under the title that is gut commensal derived succinate impels colonic inflammation in ulcerative colitis. So in this talk, I will cover the following question: how elevated succinates correlate the severity of the disease, how we can block this axis to ameliorate the IBD, and how we can design the novel consortia to engulf the accumulated succinate so that we can ameliorate the disease. So now, before going to the main course, I would just give the brief introduction. What is the IBD? So in IBD, actually, as per the clinician view, it's like there are lesions in the colon, and when we will extract these colons and we will do the analysis of the immune cells, there will be the cells there are of 2 types, the disease promoting cells and the disease preventing cells. So particularly in disease, when the dynamic will change to the pathogenic side, some more of disease promoting cells will be there. But if these cells are highly plastic, they can be converted back into the regulatory cells. But we need the modulators that can convert back into the regulatory cells. You know, the data is known that in the gonto mice, without microbiota, the maturation of Treg cells will never take place. So it means there is a potential to convert back into the regulatory cells, that is into the gut microbiota. So as per the microbiologist view, there are more than 100 trillion microbes are present. And these are the microbes, or they have as per the nutraceutical experts view, what they are doing and how they are doing with the body, they are communicating with their signals in the form of the metabolites. They are combining together with the microbiota, and they are making the microbiome, and they are communicating with the immune cells through their sensor, and they can change the outcome of the disease that may increase the disease, and that may decrease the disease. So in that form, then how these metabolites are sensed by the immune cells, because one of the most abundant metabolites in the body is the short chain fatty acids, and that is sensed by a range of the GPCR coupled receptors. They are present on the immune cells. So particularly But how, but we need a model, experimental model, how we can create that model that more of the T cell will come into the picture and we can see the sensing between the metabolites and the T cells. Because in DSS model, that is more of the barrier function studies, but this is the hapten-based model that we channelize the cells, particularly TH2 and TH9, that will mimic the UC conditions with the humans. So in that particular case, we did the experiment and we just compare both the compartments of the UC patients in the experimental mice and with the human serum also. Among both the compartments, we found one of the key metabolites that was highly upregulated, and that was the succinate actually. We correlate with the sensing receptor that was present on the immune cells, pathogenic cells that was highly upregulated. It means both are in the picture, both are simultaneously present. So after that, we did the opposite of this experiment. We put the succinate from the outside. But when we put the orally, it is not accumulating in the gut colon because it is very small in size and got absorbed from the duodenum and all that part. But we need something that can locally increase the level of the succinate. So what we did, so in that case, we did the enema, more than 15 millimolar of the succinate, and we found a similar kind of pathology in those conditions. After that, we start thinking that how we can work on these nodes. Either we can work on the iron, we work on the receptor, or maybe we can work on the succinate, and that can ameliorate the IBD condition. So after that, we just did the first experiment. We did the neutralization of the iron, and we got that there was a decrease in the pathogenic cytokines levels or increase in the healing cytokines like IL-10, that is the anti-inflammatory cytokines. And there was amelioration in the IBD clearly. After that, we designed a blocker of the receptor that is with our collaborator, and we used the blocking of this receptor in the cells, and we got the same result. Expression of receptor is going down, and there was pathology was gone. So in that case, but why these particular cells are sensitized more? Because they have more expression of this particular receptor, the TH2 and TH9, because they are the major source of IL-9 in our body. After that, we start thinking that blocking of these two, we can ameliorate the IBD condition. But what about the succinate? So in succinate, we start thinking that either first we have to find out the source and how we can block it. So after this, we did the experiment in the pseudo-germ-free mice, where we found that without the microbiota, there was no increase in the succinate level. After that, we did the FMT experiment. In those FMT experiment, in the disease one, there was a transfer of more succinate producing bacteria in the gut, and there was elevation in the pathology as per that. So we can confirm. After that, we confirm the source. Actually, we found that vulgaris is the bacteria that is the key producer in the gut. And we also colonized this bacteria, and we checked the level of succinate that was high. After that, we correlate with the human settings also. We found that there was more Bacteroideaceae and less Acidomycococci. Acidomycococci is the consumer family and Bacteroideaceae is the producer family. So there was a disbalance of the ecosystem in those settings. After that, we start thinking that we got the source, this is the gut microbiota, but how we can block it? Then we start thinking that we can use something that can block the producer, or either we can increase the number of the converters. So we work on the vulgar test, and we found that there was a retinol sensor was present, and we put the retinoic acid, it will lead to the death of the vulgar test, and levels of the succinate will be gone like that. And we put the similar experiment, and on the basis of this information, we design a consortia that is producing retinoic acid in the gut. And we colonized this, and we did the similar experiment. We found that there was a decrease in the succinate along with the decreased amelioration of the condition. After that, we used the converter bacteria that can convert the succinate more into the short chain fatty acids. After that, we did the similar experiment, and we made a consortia using these two, and we colonized them. We tested in our setting, that is, they are working fine. They are decreasing the level of the succinate and all that. And overall, the summary is like, due to the dysbiosis in the IBD, there is a dysbiosis that is leading to the disbalance of the bacteria, and that is leading to the more production of the succinate, more of the proliferation of the pathogenic cells, and that leading to the more of the IBD condition. But by blocking these states, we can decrease the amelioration. Sorry, we can promote the amelioration of the condition. And finally, I would like to acknowledge our team at Teachers TI, my boss, and all our collaborators. Thank you. I'm ready for the questions.
Thank you very much, Radip. We can have one or two quick questions. It's very good information, the role of succinate, I believe.
Yeah, we got the patent.
So genera, species, and quantification also?
Yes,
How
yes.
much, which one?
Yes, all together actually.
And this is all lab-based?
Yes, we isolated these strains from the human settings.
For GLP?
No, we are planning for that one actually.
You'll have to plan?
Yeah.
Okay, all right, good.
Any other?
Okay. Thank you.
Okay, thank you,
Thank you.
Radip.
Thank
I have a question.
What enzymes did you use to break the succinate to short chain fatty acids? What enzymes did you use to break succinate to fatty acids, more smaller fatty acids?
Not enzyme exactly. We use the consortia, bacteria.
Okay, the same.
They are the
concept.
converter. Actually, in a healthy condition, it is like that there is a balance of the ecosystem. There are producers and consumers. And succinate is short-lived, because not because of its chemistry, because of its demand actually, because it is the precursor for the making of the short-chain fatty acids. But if there will be no converter, then it will start accumulating. If once get accumulated, then through the SLC13 transporter, it will reach to the mucosa. And that will reach to the receptor carrying cells. They are pathogenic cells, and they will proliferate more. They will proliferate more cytokine. That will be the pro-inflammatory, and aggravating the disease will be like that.
Okay, thank you, Rajdeep.
Yeah.
Next.
Hello. I will now invite Mr. Akash Kumar Kharwar. He's from Naipur, Hyderabad, and he's going to talk on L. plantarum and L. helveticus and propionic acid.
Good morning, everyone. So I would like to say thank you to the organizing committee to give me chance to present here. So the topic of my presentation is Lactobacillus plantarum and helveticus elevates the propionic acid and valproic acid generated autism spectrum rat model. So first, I would like to introduce about autism. So it is a neurological illness growing up day by day. It's increasing. It's around last past 30 years is 39%. So autism is characterized by stereotype behavior in which repetitive section we have seen, and less interested in the social communications. So in autism many characters are there, like environmental factors, genetic factors, and metabolic factors. So we are focusing on this gastrointestinal dysfunctions, in which leaky gut syndrome we have found. So in current market scenario, we have seen that the probiotics holds the promise, the potential therapeutics approach addressing the GI dysfunction in autism. So these are the prevalence rate of autism, in which you can see that the map in which darker one have more autism, and if you see the Indian map, you can see the prevalence rate of autism disorder. The scale is measured by the prevalence per lakh of population. So the rationale of this whole study is to check the different animal model help in studying autism like behavior, and this model allows the exploration of probiotic in ASD related phenotype and the mechanism. So in autism condition, dysbiosis happen. So this dysbiosis is further created like a neuronal alternation and this metabolite alteration, and with the help of this probiotic, in which we use like plantarum and helveticus in our lab, we have done many of the studies like with different lactobacillus species like rotary, paracasey, but with the help of these two combination, we have found that this alteration happen in ASD condition with by this dysbiosis is balancing by with this combination of probiotics. So the main objective of this project is to investigate the impact of this both probiotic in propionic acid and valproic acid generated retin. So this is the model we used for this autism. This first model is PPA model, that is propionic acid model. Second one is valproic acid model. So these are the result in which these are the behavior results. So first we have checked the repetitive and stereotype behavior with the help of open field test, and in this result you can see that the probiotic rescued this repetitive and stereotype behavior, and we have done this grooming test in which we have found that the reduce in the episodic of this grooming in treatment group. And this is the another behavior test which is MBT's marble burring test, in which we have found that the treatment of this both combination reduces the burring efficiency of this autism, right? And then we go for the anxiety related symptoms in autism. So with the help of this two combination of probiotic, we have found that the significantly reduce in the anxiety. And this is the another behavior test in which we have checked the social interaction of this autism rat. So we have found that this probiotic have efficiency to improvement in the social interaction of this autism rat. And then we done the molecular part like pro information. We go for this pro information markers like IL-6 and IL-6 and TNF alpha, and with the help of this both probiotic combination, we have found that the increase in the pro-inflammatory marker in the autism rate is decreases by using of this both probiotics combinations. And another, we have checked the oxidative stress in the plasma level in osteotic rat with the help of these two tests of SOD activity and catalysis test. Then we check for the metabolites with the help of the NMR in fecal sample. So we have found that the metabolite which are decreases with in autism through dysbiosis is restored with the help of this using this both probiotic. And after that, we have seen the morphologically in valproic acid model, this probiotic improves the intestinal morphology of this colon. So then we go for this tight junction protein which are disturbed in autism through this dysbiosis. This both tight junction proteins are Z1 and occludin. So this probiotic all have effect on the restoration of this Z1 and occludin in autism rat. And we have checked this neurotransmitter like dopamine and GABA, which are decreases in.
Akash, come to conclusion.
So so overall the conclusion of this study is VPA rat exhibit autism like behavior which is mimicking core feature of autism, and this both lactobacillus plantarum and helvanthichus elevate the stereotype and anxiety behavior by enhancing the behavior function in gut microbial balance. Both probiotic reduce this pro-inflammatory marker and restored the neurotransmitter in PFC region. Okay, thank you.
Thank you. Any question?
Pardon.
Any strong scientific rationale for selecting these two species?
Ma'am,
Lactobacillus?
yeah, ma'am. So in our lab, we have checked for this rotary and this Paracasey and other lactobacillus also. But with the combination of this both plantarum and helvanthichus, we have found that the restoration of this pro-inflammatory and short chain fatty is more as compared to both. We used single strains, like done single strain study like plantarum, singly plantarum and then helvanthichus. So these both show some concerta type of actions.
Okay, thank you very much, Akash. Now next, I invite Anand EP from TCS Research Pune.
Hi, good afternoon. Thanking the organizers for providing me an opportunity to present my work here. So the work is titled Knowledge Graph Guided Personalized Probiotic and Dietary Recommendations for IBD. First of all, briefly introducing about IBD. IBD, as we know, is a multifactorial and complex disorder, which is influenced by the interaction between host genetics and gut microbiome. And this results in abnormal inflammation and, as depicted here, changes in gut barrier integrity, especially the disruption of gut barrier. And modulation of gut bacteria through factors like diet and probiotics. It can be a very effective solution when given in a personalized manner, and it could possibly help alleviating IBD symptoms. And several computational tools can be used to design these individualized approaches. Knowledge graphs is one such technique which can capture elegantly the heterogeneous interactions which happen in a complex disorder like IBD. So, going to the objectives of this work, firstly, the objective is to construct a gut bacteria-centric knowledge graph with a focus on gut bacteria to host associations. And secondly, developing a score that reflects the association of each bacteria with various host components that are reported to be involved in IBD. That is, factors which contribute to the development of IBD. And thirdly, to develop a workflow to design a personalized dietary and probiotic recommendation using microbiome data from individuals with IBD. So in methodology, we created a knowledge graph, which is basically a collection of all kind of association data in the form of triples or a subject, object, and a relationship between them. So the nodes involved in this knowledge graph include bacteria or gut bacteria at the center and host-related factors like host gene, diseases, food, and bacterial factors like other interacting bacteria, which could be probiotics, and bacterial functions, etc. So this knowledge graph was populated using data from curated public databases and also from patterns or data from experimental outputs from peer-reviewed journals. And biomedical text mining was also used for this purpose. And we have recently published a work in which we have given a methodology which can be used to take out interbacterial association data. Now, suppose we have a data from an individual, the clinical data as well as a microbiome profile. This data, we can see here, we have a list of bacteria which have been obtained from the person's gut. And the knowledge graph that we created called GBKG can be used to overlay on this list of bacteria so that we can create a matrix where we have a list of bacteria as the rows and we have all IBD associated host factors as the columns. So this could be IBD associated genes, immune factors, chemicals, and diseases. By diseases, I mean if there is any known association of the bacteria to ulcerative colitis, CD, or any other variations or different kinds of different stages of IBD. Now, this matrix can be populated again. So we see that this would be a very sparse matrix because most of the associations may not be available, or very few associations must be reported in the literature. And we generate another matrix using a method called collaborative filtering, where we try to fill in all values here. So basically, with this, these will be the predicted values, which could denote the association of bacteria to those factors. And if we sum all these values across all the rows for each bacteria, we can generate a score, which is called a gut favorability score. And if we scale it between 0 to 1, this would mean that a bacteria with a value closer to 0, GFS score closer to 0, would mean that it has low association to IBD associated factors, and a value closer to 1 would mean the association would be larger. Now, this association data and food association data from GBKG can be used to recommend food as well as probiotics. So in the first phase of recommendation, we can directly query the KG and use the GFS data to get a list of food and probiotic. And later, it can be modulated based on the person's preference, calorie, etc. So in results, we have constructed a very extensive KG, including a lot of diseases, and it's a broad KG. It is not IBD centric. And in this KG, we have nearly 25 million triples or edges and around 6 million nodes with 80 relationship categories. Now, this is an example graph which we obtained after querying based on representative bacteria. And this is how the relationships will appear in a graphical way. So we have a sample and we obtain the microbiome data, and after overlaying on GBKG, this is a representative graph. And in each of the bacteria in this will be given a gut favorability score, which can be used to give a recommendation. And for example, this is a sample recommendation which can be generated with this. So basically in this work, we present GBKG, a knowledge graph, which is a very flexible tool, and it can incorporate gut bacteria association data and later can be developed to incorporate much more data. And using this, we develop a favorability score incorporating different host components that are associated to IBD. And clinical validation is, of course, necessary at each and every step of this. And this methodology can be actually incorporated into any mobile-based application, for example, to obtain more personalized clinical data of the person. And a combined recommendation of diet as well as probiotics, which could make use of this GBKG at some level, would actually be a good strategy that towards the treatment of IBD.
Thank you.
Thanks.
Any question?
I have one question. Actually, you have so many combination permutations because different type of diets and foods and nutrients and different type of bacteria. So how you will decide that which criteria will be in priority and which with lower priority like that?
Yeah, so basically we can do a weightage like that, but in this case, we just provided a basic methodology. And we are not specifically giving any weightage to any of those
Is it
criteria.
targeted towards specific application, maybe for IBD or maybe for immunity or like that?
So basically, in this context, we actually created a very broad KG, and parts of it, like you can target maybe a very specific disease, or you can target all diseases associated also. So it is up to the user. The user can query for a disease, like all the factors associated to the disease, and they can get it out of the KG and then use it according to whatever situation they are in. So that is in the importance of this kind of KG is that it is usable in multiple contexts, and this technology can be adopted to disease specific or broader way also.
Okay. Any other
Okay,
question?
I just want to ask a question here.
Yeah.
Yeah. So I think the concept is brilliant. You're trying to put all the variables together.
Yeah.
My query is from where have you taken the food database?
Okay.
And how are you arriving at the, recommendations for the diet? Because I saw in the last slide, some recommendations were made. Can you please elaborate?
Yeah. So actually the availability of data is very low regarding exact bacteria to exact food correlation. So we had searched extensively about that, and we found a few literature evidences. So the sample data, what we showed here, was basically from that data. So it was just, I think, 2 sample papers, which had kind of extensive data. And in that, the association to foods were at different levels also. So some of the foods were like very specific, like for example, a bacteria is known to be associated positively with red wine, for example. But there could also be associations like bacteria is known to be associated to group alcohol in a positive way or a negative way. So recommendation there is actually a bit tricky. So ideally it would require much more data, much more extensive data So
it
we need to
cannot,
collect
it cannot be just food. It has to be the component of food.
Yes, yes. So we
Because
tried
when you
this.
talk about diet and
Yeah.
IBD, you know, it has to be more specific and precise.
Yeah.
So
So we did this manner also, like a bacteria is known to consume a carbon source or a nitrogen source or any such material. And this is a part of a diet or a food. And we had tried to build a relationship through that also. But the problem here is also that not even if a component is present in a food, it may not be necessary that the bacteria will utilize it at that moment in that patient, in that particular stage of the disease. So that is a very difficult problem to do. So obviously the recommendations, whatever we make through it, have a lot of context which is required. And one beauty of knowledge graph is that there are different graph databases in which we can load the data. So in some of the graph databases, for each relationship, suppose for example, Ruminococcus navis, we know it is related to MUC1 gene. So, and that we know it is upregulated in IBD. And this is a relationship which we have. And this relationship can be represented.
Okay, Anand, thank you. We understood. Thank you very much. Now, next is Mithali Inamdar, National Center for Cell Sciences, Pune.
Hello. Yes. So very good afternoon, everyone. I'm Ms. Mithali Namdar. I am from National Center for Cell Sciences. I'm working with Dr. Dheeraj on mother-infant microbiome, and today I'll be presenting the same. Thank you, organizers, for giving me this opportunity. To start with, we all know that microbiome is an integral part of our body, and especially the infant microbiome is very, very important because it helps in development of various systems like gastrointestinal tract, the neurological as well as immune system. And ultimately, it helps in deciding the fate of our body. But any dysbiosis during this early acquisition of microbiome may lead to obesity, diabetes, colitis, or certain severe disorders or diseases. So this infant gut microbiome is affected by various, or there are many factors which are affected in the development of infant gut microbiome, is like the mode of delivery, the mode of feeding, etc. And there are various intrinsic factors also which help in development of this infant gut bacteria. For example, the HMOs. So human milk oligosaccharides are nothing but small sugar moieties which are present in the breast milk, which are not digestible by human body, but only digestible by infants. And which when digested by infants, these beneficial bacteria are proliferated, and that in turn leads to secretion or production of HCFAs like acetate, propionate, and butyrate. So I won't be showing this data today, but understanding the importance of mother-infant topic, we designed a project which initiated with the sampling of mother-infant cohort population. So we sampled 40 different mother-infant pairs, which resulted into 760 samples across 4 different time points. And we collected samples from mother's stool, skin, and breast milk. And from infant, we collected stool as well as oral swabs. So for all these 760 samples, we have done amplicon as well as shotgun metagenomics. This is the following workflow which we followed for amplicon sequencing. And I'll be showing you the results for amplicon sequencing now. So the initial results showed that the alpha diversity across different body sites in mother and infant were varying, and especially the mother's gut had the highest diversity. And we got the but obvious result that the infant gut microbiome is populating or is maturing over the time, which we can see in the alpha diversity. But the most interesting part which we saw was in the beta diversity. So here you can see that different colors are denoting the different body sites of infant and the mother. And you can see that the mother's gut and the infant's gut are having similar kind of diversity, which is shown in the beta diversity plot. So this really was an intriguing result in our analysis, because how do these gut bacteria are populated in the first time point itself in the infant? That means just after the birth. So you must have heard about yesterday's talk given by Professor Saurabh Datta that there are different ways in which infant gut is populated. So there are hypotheses like intraplacental, intrauterine, as well as the third and the most interesting one is the enteromammary pathway, which means in the enteromammary pathway, there are certain bacteria which can be seeped out from the gastrointestinal tract of the gut of the mother and get populated with the immune cells like dendritic cells in the breast milk, and which when fed internally or which when fed by infant are then populated by infant gut. So we started questioning, and then we further led to different kind of analysis to see whether this is really happening. So we did a different kind of analysis called as cosine similarity indices. So in this, we have kept as infant gut one vector and another vector different body sites of infant and mother. And we saw that in the mother gut, so the infant gut was having a highest similarity with that of mother's gut across time point. Again, the same point was getting showed from the previous results itself. Then we further moved to source tracking. So we use source tracker 2 here to see what are the sources of infant gut bacteria, and is mother samples or are the mother microbiome contributing in it. So we can see here again that the mother gut has the influence, followed by the breast milk bacteria, which is there in the infant's gut at the time point 1, and eventually the breast milk samples contribution is getting lowered and the mother's gut is getting increased, because you know that the infant's gut is also maturing. So taking hints from this, we further did the shotgun metagenomics, and there we found that the bifidobacterium strains are the most abundant strains which are seen in all the 3 samples. That means in infant gut, milk, as well as in mother gut. And then further, we took these mags and we did the strain tracing of these bifidobacterium strains, and we saw that the bifidobacterium bifidum, if you can see in the pink color, and the bifidobacterium longum are the 2 strains which are transmitted from mother gut to the milk to the infant gut. So now we know which bacteria is actually getting transmitted. But there are certain drawbacks to this study. And so these studies are basically the molecular based studies and usually done based on DNA technologies. So there can be a chance of contamination, or there can be certain hints that there can be a contamination. So we further decided, so I want to refer to yesterday's talk for this, that there are different kinds of pathways. So if we further did isolation of bifidobacterium species from mother's gut sample, and we did transformation. So we transform M. cherry, that is a fluorescently tagged, red fluorescence protein, and we tagged it in the bifidobacterium, and we did animal studies. So in the animal studies, we gavaged this M. cherry tagged bifidobacterium from the pregnancy of the mice till the delivery of the pups, and we checked for the translocation of this bacteria. So there are different methods which we approached for checking this bacteria. So we did IVIS imaging. So IVIS is a whole body imaging. We did qPCR. Yes. So we did certain animal experiments. We found good results also. And the results are that from the infant's gut microbiome from birth is getting populated, and there can be also enteromammary pathway in the mother-infant dyads. But the last thing which I think was very important, the significance of the study, that I would again like to refer to yesterday's talk, that is Professor Patricia's talk, that she mentioned that there are various benefits which are there in the probiotic, but in the mother-infant population, which is very sensitive, there are certain drawbacks which are being seen in the recent studies. That is, the protocols are not standardized, the strains and the dosage is also not standardized. But I think if there is any mother-infant translocation, that this can be a very promising method through which the probiotics can be delivered, because the dosage of the strains cannot, we don't need to decide the dosage of the strain. Mother itself physiologically will decide the strain, dosage of the strain.
Thank you.
Thank you.
Any question?
So PYG plus blood plus rumen.
And you do the spread plate, right?
Yes, we did enrichment and spread plate.
When you kept in the anaerobic chamber.
Yes, we have used Don Whitley 835.
So if there is no question, thank you very much, Mitali.
Thank you.
Thank you. The now last speaker is Sucheta Guleria from PGIMER, Chandigarh.
Good afternoon, everyone. The title of my study is Giardia infection is associated with altered community structure of murine gut microbiome post antibiotic and probiotic treatment. So starting with the introduction, giardiasis is a gastrointestinal parasitic disease which is caused by a protozoan Giardia duodenalis in humans. It has two forms, that is the trophozoite form and the cyst form. And the cyst is responsible for the transmission. And once the cyst undergoes excistation due to the gastric secretions in the stomach, the trophozoites, they attach to the intestinal epithelial cells and they cause various gastrointestinal symptoms. So giardiasis is associated with acute and chronic diarrhea in children and adults worldwide, and also result in post-infectious complications, that is irritable bowel syndrome and chronic fatigue syndrome. And interestingly, 75% of the infection is asymptomatic. So the exact mechanism behind giardia pathogenesis remain incompletely understood, but recent research underscores the crucial role of the microbiota. As we already know that the intestinal microbiota, it plays a critical role in the gut homeostasis and overall health, and its alteration by the entropathogen is associated with the gastrointestinal diseases. And also the dietary interventions, they contribute to the microbial diversity and it influence the giardia pathogenesis. Therefore, understanding the role of specific commensal bacteria during the infection could help in exploring new treatment options and monitoring therapies such as fecal microbial transplant. So the objective of my study is to evaluate the shift in the community composition of the gut microbiome associated with giardiasis in untreated and pre-treated, that is pre-treated with antibiotics and probiotics in mouse model. So coming to the material and methods, so the study includes 168 biopsy mice. They were further divided into 3 groups. The first group was treated with antibiotics for 4 days by oral gavage. The second group was treated with the Lactobacillus rhamnosus, that is the probiotic, for 7 days by oral gavage. And the third group was not treated with antibiotic or Lactobacillus. Now each group was further divided into 2 subgroups. The one subgroup was infected with the Giardia trophozoites, and the other subgroup acted as its respective uninfected controls. And mice in the replicates of 8, they were sacrificed at 3 time points, that is the 3, 7th, and 14th day post-infection. So for the Giardia infection, we have maintained the azeni culture of this parasite in our department. In the TYS33 media, you can see the culture tubes. These are the tubes in which the parasite is maintained. And the figure also shows the trophozoites under the 10x magnification and the trophozoites in the iodine as well as the wet mount under 40x magnification. This is how we have given the animal inoculation of the Giardia trophozoites as well as the probiotics. And the Lactobacillus rhamnosus has been maintained in the MRS agar plates. So for the assessment of the microbial diversity, the luminal contents were extracted. They were isolated, DNA was extracted, and the V3, V4 region of 16S was amplified. And also to actually study the impact of these pre-treatments on the Giardia trophozoites, we did the quantification using the quantitative PCR. So coming to the results, as we can see from the first diagram, the swelling of the large intestine actually shows that the antibiotic lead to the dysbiosis of the commensals. And to confirm the establishment of the lactobacillus infection, we performed the lactobacillus specific PCR. And to confirm the establishment of the Giardia infection in the mice model, we actually performed the nested PCR targeting the TPI gene of Giardia. So coming to the microbiome analysis, we can see that the antibiotic pre-treated and untreated group, they lead to the increase in the phylum Proteobacteria, Campylobacterota, and decrease in phylum Firmicutes up to 14-day post-infection, in contrast to their respective uninfective controls. Whereas in probiotic pre-treated group, it leads to decrease in the phylum Proteobacteria and increase in the phylum Firmicutes up to 7 days, and decrease in phylum Campylobacterota up to 14-day post-infection, similar to respective uninfective controls. So as you can see from the alpha diversity, the antibiotic pre-treated group, it basically remained consistently lower at all the time points, indicating that the dysbiosis of the antibiotics along with the GRD acid, it further disrupted the commensals as compared to the untreated ones, whereas the probiotic treated group, you can see the alpha diversity is higher at all the time points, indicating that the integrity of the composition of the gut microbiome, it was intact regardless of the Giardia infection. From the qPCR analysis also, we can see that the Giardia infection actually led to the infection intensity, increase in the infection intensity at 3rd day, peaking at 7th day and further decline at 14th day. But when we compare to the antibiotic pre-treated group, we can see the 10-fold increase in the Giardia quantification. And on the other hand, treating it with probiotic, that is Lactobacillus rhamnosus, we can see the gradual decrease from 3rd to 14th day. So coming to the discussion, as I've already told that the Giardia duodenalis, it leads to the increase in phylum Proteobacteria, Campylobacterota, and decrease in firmicutes up to 14th day, whereas Lactobacillus pre-treated group shows the opposite trend with increase in Proteobacteria and firmicutes, but only up to 7th day post-infection. I wanted to highlight a point here that it leads to the contrasting results only up to 7th day, suggesting that the regular dose of probiotics are required for maintaining the gut flora, whereas the Shannon and Simpson diversity indices, they were also consistently lower in the antibiotic as well as untreated mice, whereas the probiotic fed group, they exhibited the highest diversity, and it basically suggests that regardless of the Giardia infection, the microbiome remained intact at all the time points. And moreover, from the RT-PCR analysis, the antibiotic pre-treatment exhibited a robust infection rate, and the lactobacillus administration, they reduced the parasite burden at all the time points. So basically, the study found the potential danger of the antibiotic usage causing the greater disruption of the gut microbiota as depicted by the alpha diversity, and resulted in more severe and prolonged Giardia infection as compared to untreated group, whereas the mice pre-treated with probiotics, the microbial diversity remained intact regardless of Giardia infection. Also, it led to the reduction of the parasite load, mitigating both the severity and the duration of the infection. So key message, the study emphasizes the importance of the intestinal microbiome against Giardia infection. These findings suggest that antibiotics usage during this infection could lead to negative health outcomes, and the study provide potential avenues for the probiotic therapy and help in monitoring strategies such as fecal microbial transplants. Thank you.
Thank you. Any question?
Yeah.
Yes, please.
By not using the right antimicrobial. Your study said that in the first group you use ampicillin, neomycin, and I don't know what all.
Vancomycin,
Vancomycin.
yes.
For Giardia treatment is using metronidazole, tinidazole, ornidazole, or you have the new one, which is nitazoxanide. So why did you use this group? Because you're starting with treatment with antibiotics for a disease which is not going to work.
Actually,
and then you compare it by giving probiotics and say that this is the best way, the results are good. So that's why this
Sir,
is the point.
actually, the metronidazole and tinidazole are the drugs for against giardiasis, but as it causes diarrhea, that is acute to chronic diarrhea, it is misdiagnosed and people take antibiotics for diarrhea. So to cause the dysbiosis in the mice model, these particular antibiotics, they were used in order to
I'm saying if you had used metronidazole, it would have given the result which would have been comparative, that whether the probiotic is as effective as metronidazole or not.
Yes,
Here
so
you
we can
compared it
add
only
that
with
one group also of the drug also.
Okay. If there is no other question, I think I'll invite jury to declare the results.
Thank you.
Thank you so much. So all 5 are very brilliant presentations because they have been screened at 3 layers. And now we'll go for the other formality. Please.
Let's have a huge round of applause for all the young investigators. Now, may I request Mr. E.G. Amano to felicitate our chairperson, Professor Jyoti Tamang. Can we have a huge round of applause for our chairperson, Professor Jyoti Tamang? And also I request Mr. Tomoyuki Iwama to felicitate our chairperson, Professor J.B. Prajapati. Let's have a huge round of applause for our chairperson, Professor J.B. Prajapati. Let's have a huge round of applause. Thank you. We now move on to the one of the most exciting part, the panel discussion, which will help us understand and address some of the questions that often come to mind about the future direction of gut microbiota and probiotic science. We would now like to invite Professor B.S. Ramakrishna, Director, SIMS Institute of Gastroenterology, Hepatobiliary Sciences and Transplantation, SRM Institutes of Medical Science, Chennai, India, to moderate the panel forward and introduce our panelists.
Invite our panelists. Dr. Shashikaran, former director of the National Institute of Nutrition, Hyderabad, and a key member of the food FSSAI, and particularly involved in nutrition. I think we'll have Dr. Shashikaran's bio data up here. Dr. Prakash Halami, Chief Scientist and Head, Microbiology and Fermentation Technology at CFTRI in Mysore. Dr. Ajay Balla, Director and Head of Gastroenterology and Hepatology at the Fortis Hospital in Noida. And Dr. Sharad Gopalan, Senior Consultant Pediatric Gastroenterologist and Hepatologist at Rainbow Children's Hospital in New Delhi, and the President of the Nutrition Society of India. And Ms. Ishi Khosla, a clinical nutritionist and an entrepreneur, and the founder of the Celiac Society of India. So this panel discussion is titled Future Directions of Gut Microbiota and Probiotic Science. Can I have the slides, please? Yeah. Can we have it on full screen? All right, and you've seen the panelists here, distinguished panel out here, to talk about what the future is like. So some things we didn't have 25 years ago, some things which have happened in the 25 years, we have food delivery, anything that you want at home in 10 minutes. You can look at your phone and navigate your way across the world, anywhere, wherever you're in the world. In most places, you can do that. That's something we didn't have 25 years ago. You have joint replacements, this medicine. I mean, you can see this person doing yogasanas. And joint replacements have become so commonplace, even at extremes of age, that it's really made a huge difference. And we have, in the last year, heard that rectal cancer can disappear with medication, totally. So these are things, the world is really moving very rapidly. And the question that I have to the panelists will be that please look into your crystal ball and tell us what you think the world will be like in probiotic science in 2050. That's 25 years from now. So the regulatory, so I'll start with Dr. Shashikaran. So we'll have a round of questions, and if we have enough time at the end of it, then we might come back with questions from the audience. So we know that the regulation currently depends on fairly stringent standards in terms of production, in terms of what is included in a probiotic product, and health claims. So what changes do you see, Dr. Shashikaran? Will regulators allow things like genetic characterization, metabolomics, and computational methods? I mean, we heard a lot about AI today, even in this young investigator session. Will they use that to decide health claims? And is there, we have national standards. Are there going to be international harmonized standards for regulating probiotics? And how will the regulatory bodies look at personalized medicine?
Yeah. Thank you. And they're extremely interesting questions, because the one thing which moves the slowest in our country is regulation. So when the science is moving fast, the regulation rarely keeps pace with it. And as you have said that 25 years from now, what regulation will be is, we are hoping that it will be something better than what it is today. The present day regulations were based on ICMR guidelines prepared on probiotics, which we released in 2008 under the leadership of Professor Ganguly. That was the basis for the regulations. But the regulations were not foolproof, and we already have a lot of issues regarding several aspects. So now, when it comes to changing regulations, it is usually done by a group of scientists, and they look for a lot of evidence. And based on the evidence, only the regulations are made. Now, whether it's based on genetic characterization, metabolomics, or computational methods, etc., it again depends upon how the panels actually view what is called as evidence. At the moment, the evidence for any regulation is based on scientific studies or clinical studies. So usually, basic data or animal-based data are rarely used for framing regulation. And therefore, very often they insist on clinical studies being done to generate that particular kind of regulation. And especially when the product is making a claim, a health claim, then there is definitely needs to be a clinical study. Even all the genetic, computational, etc. information will only form some background evidence. The ultimate proof of the pudding is in the eating, and therefore the human study a well-controlled, well-designed study becomes imperative. And then, of course, will it be harmonized with international standards? Yes, it's already, we have harmonized with Codex standards. The Codex is the international standard which all countries agree upon. If there are no Indian standards, we could also generate our Indian standard. For example, RDA for vitamins. If we were to have an RDA for probiotics, or RDA for prebiotics, and then the regulator will adopt the Indian RDA. Wherever that kind of information is absent, then we go for the Codex regulation. If the Codex is also not there, then it's entirely based on the published evidence of that particular day. And the last one, of course, is whether will probiotic, biotherapeutic products be licensed for personalized medicine. In fact, the regulation does not have any role on personalized medicine. Regularizes only for a generic one, which applies across the country. So personalized medicine, of course, it would have said entirely on the clinician or the dietitian to devise that, but it should anyway fit into a regulatory framework, which today doesn't exist.
So will personalized medicine then go to the drug controller?
It might. It is more likely it will go under the drug control rather than the food-based.
Okay. So thank you, Dr. Shashikan. And I'll probably move on to the next one because we'll come back to these things. We already heard a little bit about postbiotics. In fact, so the initial sessions today also had things about postbiotics in terms of microbial products being used rather than live biotherapeutic products. I'll call on Dr. Prakash Halami to enlighten us exactly on what are postbiotics. Are there postbiotics in commercial use currently, and where do you see them in 2050?
Thank you very much. Thank the organizer for inviting. About the postbiotic, in fact, the talk by Mr. Kato made my job easier. Here, Lactobacillus casei, the exopolysaccharide producing culture, has a space resistance. So postbiotic actually has been defined in 2019 by International Scientific Association for Probiotics and Prebiotics as a preparation of inanimate microorganisms and are their components. This will exhibit the health benefit. So these postbiotics can be from the gross microorganism or the probiotic organism, inactivated microbial cells, metabolites produced by them, components of the cell wall, then cell lysates, vitamins, and of course, which we are seeing the short chain fatty acid, cell-free supernatant, and more commonly used is the exopolysaccharide. This actually comes under the postbiotic molecule, fruit preparation.
One of the very basic ways in which probiotics work is through the innate immune system, right? And so innate immune system just senses different components of either the bacterial wall or some bacterial nucleic acid or something, depending on which receptor it goes to. And this is really what modulates immune function. So are any such products, let us say bacterial nucleic acid or viral nucleic acid, available currently in commercial form? Do you foresee that coming into clinical use?
Exactly. There are actually such a 4 products which I could see in the literature. And most of them are actually patented. One is the EpiCore, a Saccharomyces cerevisiae metabolite, which is exclusively used as a food supplement. And this works for reducing the intestinal symptoms as well as the mucosal defense. This has actually the constituents like protein, fiber, polyphenols, antioxidants, vitamins, and the polysaccharide which they have reported is the beta glucan and manospecific. This is actually being sold in the market, available actually in the market. Another one is actually Beta Via, a Kemin company which actually sells, and this is from the algal based. Again, this is also the beta glucan. The algae is the Ingena gracilis, and this works actually for the immune system, the gastrointestine, as well as increasing the good microflora also. Help for this healthy gut barrier and again the urinary tract infection. This is being sold by Kemin, and then Epicor is actually available also as the product. There is another formulation by Edims, ECI, EC1. And EC1 actually is the lysate of Bifidobacterium longum and reported for the gut health and used for IBS system, as well as reduces the TNF alpha and take care of Clostridium difficile infection. This is just a inactivated cells of Bifidobacterium longum probiotic. The last one I came across with one formulation is cytophrina, comprises of the lactobacillus and then Bifidobacteria. This is being sold by I heard a probiotic as a probiotic lysate tonic, useful for the intestinal dysbiosis as well as strengthening the immune system and known to promote the beneficial bacteria, and also has a function for the bowel regulation. The last part, which is shown there in the screen with reference to what will be the postbiotics market after 25 years. So we see by 2031, the postbiotics will be almost the market size of $3 billions. And presently it is with a compound growth rate of 6.8. India actually has a very good jump from 14.8% because we use the several organic as well as Ayurvedic product as a dietary supplement. And Nutralite is one such formulation being used for the bone health and immune system. These are actually the information as of now on the postbiotic available.
So you feel that this is likely to expand significantly? Thank you. We heard a bit about fecal microbiota transplantation. Now it's everybody in the GI world has tried fecal microbiota transplantation, which is, as you know, Dr. Balla, that this is very unfocused. So the only recommended indication for it is recurrent Clostridium difficile infection. There is a role maybe in ulcerative colitis. You know that several groups are using it for their patients with UC. It's also tried in other GI conditions, alcoholic hepatitis, IBS. And then there's a huge call when people keep calling and asking, can you do this for my child with autism? In CKD, can you do it? In IgA nephropathy, can you do it? In diabetes, obesity, and so on. Alzheimer's and Parkinson's joining the list now. Now, we also know that new probiotic formulations have come. So this is a probiotic formulation which is given rectally, which is to prevent Clostridium difficile infection. It's sold as Prebiota. And then you've heard of this thing called VOST, which is an oral formulation. It is basically Firmicutes. These are spores of those which are collected from healthy people and marketed in the US. So my question to you, Dr. Balla, is do you think probiotics can replace fecal microbiota transplantation? Do you think probiotics will be used in the form of enemas, colonoscopic infusions, or oral capsules?
I think in the last 2 days, there's been enough which has been talked about probiotics, how they are going extensively into all forms of clinical medicine. It's not the gut microbiome alone. It's managing everything, your neurological part, your cardiac part, your metabolic syndrome. So probiotics have a lot of potential, and there's no doubt about it. Fecal microbiota transplant, FMT, is Currently, as per the WHO guidelines world over, it's only recommended for recurrent CDAD, recurrent CD-associated diarrhea, not for anything else. But yes, there is a lot of role of the same. But the delivery system of FMT is the tough part. It needs to first collect it from a group of healthy volunteers, and the delivery, if it has to be done through a Ryle's tube delivered into the small intestine, or through a colonoscope delivered into the colon. So the delivery of FMT is what has been the issue. But yes, with new things coming out where the delivery module can change, things will simplify. There is a huge risk factor, to select a patient ideal donor. There's nothing like an ideal donor. We have a different microbiome. The microbiome in the West is different. So getting a capsule from US imported here, delivering it rectally may not be effective because their microbiome is different. So it's going to be a difficult exercise. It's going to be more tailor-made according to your region. So that is what would happen eventually. Yes, it's going to happen, and it will take some time. And regarding other diseases, I guess trials have been done. I have also done it for alcoholic hepatitis, done it for ulcerative colitis. But yes, if you look at it, they're all based primarily because we have got some guidelines for probiotics. It's not for FMT, but for probiotics. Probiotics, are recommended even for simple things like it prevents oral thrush in elderly if it's given adequately. It's a level 2 recommendation. You use probiotics for NASH, we use it plenty. The same thing is going to overlap into the other part. Obesity management, again, it will be there. WHO has recommended a level 2 evidence of probiotics for MHE, for minimal hepatic encephalopathy. It is supposed to be at par with lactulose. So I guess FMT is going to be just an extension of the same use of probiotics, but with such careful selection of donors. And of course, we're not going to be getting it from abroad. It has to be innovative. It has to be done from our country because our microbiome is going to be different.
So the question was, do you think probiotic colonoscopic infusions can replace FMT? This year, there's a publication which shows that in some settings it can be done.
Yes.
Where do you think the world will head? I mean, that's probiotics will obviously be much cleaner in terms of handling.
Yeah, I guess it's going to be like after we get to know what is the exact mixture. If you look at it for pouchitis, there's a mixture of 8 different forms of bacteria, which if used in the right proportion works very well in these patients. Similarly, it will be, like I said, there would be more, we'll need far more work to see which are the exact mixtures. Probably will be safer. It will be safer. It may work very well. But we need to exercise caution. Yesterday, I remember taking a call about the pancreatitis part, and I asked, are you going to recommend it? And I was so happy to hear that that is one point where the speaker said, no, I will not recommend it because I do not use the same. I don't recommend it. So we need to exercise caution. Of course, use of probiotics, if you know the right strains, right mixture, would be ideal. But there's nothing the right mixture as of now, which we know about.
So a lot more work to be done out there for the entrepreneurs in the audience here. So going on to Dr. Sharad, so a lot of probiotics are now used in children. In particular, I think allergy and so on has emerged in the West as a huge problem right from the pediatric age. I'm sure in the urban areas in India also it's emerged as a significant problem. So Sharath, what do you think in the next 25 years? What changes do you see in probiotic use? Is it going to be therapeutic, preventative? Where would you use it, and what would you?
Thank you, Dr. Ramakrishnan. I would answer your question in 3 parts. Firstly, when it comes to pediatrics, we've been always focusing on specific disease entities, like do probiotics help in acute diarrhea, acute infectious diarrhea, or antibiotic-associated diarrhea, or neonatal necrotizing enterocolitis. Rather, we should probably focus on the alteration of the gut microbiome as a separate entity itself. Any GI condition, be it diarrhea, be it constipation, be it celiac disease, be it irritable bowel syndrome, be it inflammatory bowel disease, they all have two things, either chronic inflammation or dysbiosis, or a combination of both. So if you are addressing dysbiosis, when you look at any entity like treating constipation, we focus on treating the disease entity, but we also need to lay emphasis on the dysbiosis aspect and modify the gut microbiome. Now, how much hard evidence is there for the specific role of specific strains of probiotics in dysbiosis? How long should they be used? This is an area which has aroused a lot of keen interest in pediatrics, but we are at no stage of recommendations regarding the dosage. But we have certainly know that certain specific strains of probiotics do help. Most studies which have been done have looked at durations as long as 6 to 8 weeks of continued supplementation. The rationale being you modify the gut microbiome over a period of time, making it beneficial. So that is one area we should focus on, dysbiosis rather than specific disease entities. The second area is we should probably start moving away from clinical indications for probiotics to a much larger platform of potential implications of benefits in potential benefits in public health. For example, we have several probiotics. Over the past 2 years, there's been a lot of information on probiotics enhancing plant protein absorption, as well as absorption of specific minerals like calcium, selenium, magnesium, potassium, and zinc. Whereas, in fact, Dr. Bahel had made a presentation recently where he had mentioned that there was a large study done in North India and Palwal, where they fortified iron with a probiotic and found out whether it enhances iron absorption. And they found it does not. It was a large study. So when it comes to iron, there are problems. But the evidence for calcium, selenium, and other minerals is quite promising. So this is one area we could look at. Can we improve nutrient, gut bioaccessibility, and bioavailability? And by giving less, we make sure more of it is bioavailable to the tissues. Can we look at that aspect also? That is the second way. The third thing is what Professor Jyoti Tamam has repeatedly been mentioning. We have got a whole rich information about functional foods. And this is an area which deeply interests the Asia-Pacific region. Not only India, all the East Asian countries are deeply interested in this area. For example, you have foods like kinema, you have natto in Japan, you have chikongyong in Korea. The time has come probably, as Dr. Talwar also mentioned, we should start looking at clinical trials in humans, in the pediatric as well as adult population, using these functional foods. Right now, they are largely animal experiment based. But if we can translate these into clinical trials in humans, then as Dr. Shashikaran mentioned, things about regulation and all other aspects and its potential utility in clinical practice becomes even more meaningful. Thank you.
So do you think the Indian Academy of Pediatrics, somewhere in its statements, might include something on probiotics and in terms of promoting health in infants? I mean, you're talking about dysbiosis, and this goes back to all those theories of child growth, the reason for linear growth retardation being dysbiosis, and whether that can be corrected and so on.
Certainly, but the challenge here is the term health promotion is a very broad term. When we say promoting probiotics for promoting health, and we want to come out with a recommendation there, we have to be very, very clear what specific things are we looking at in terms of health promotion. So that becomes important.
So you need community trials to show that.
Yes.
So we'll move on to Ishi Khosla. So basically, I think you are interested, and we heard a bit about the leaky gut hypothesis, that intestinal permeability is at the heart of a lot of chronic ailments that afflict humankind. And so there's a lot of probiotics out there which are sold for the leaky gut. A lot of people come to me saying, I'm not feeling good, chronically, I'm depressed, or whatever. Actually, I know that you're into this and you advocate lifestyle changes and diet changes. So there are, there's sites like these, antibodies, where, probiotics to control food allergies. So what do you see in terms of not just allergies alone, but the broader field of intolerance, which we talked about earlier? Where do probiotics come in this, and what do you see happening in the next 25 years?
Well, first of all, I'd like to thank Dr. Nirja and Yakul for having me. And this is a subject of huge interest for me as a clinician. A lot of conversations have happened over the last day and on probiotics with respect to disease and the gut. But my area is about how the gut is impacted with food and how probiotics in the microbiome get impacted. And I would just like to, first of all, distinguish between the two words of allergies and intolerance. Allergies are more acute and more IgE mediated, whereas food intolerances are IgG mediated. And a lot of my patients just use the word allergy and get these tests done. So that is something which needs to be understood better. And essentially, I would just quote Dr. Alessio Fasano. He is the chair of Boston Medical Center. He's a pediatric gastroenterologist. Over 1,000 papers he's published. And he has said it very, very, very strongly that all diseases, almost 300 medical conditions, are connected with the leaky gut. And one of the biggest contributors to the leaky gut is wheat protein. He said it very, very, very plainly in a publication. It's in, I think, published in Nature and the Journal of Gastroenterology a few years ago. And this is something which I personally also see in my practice. And use of probiotics along with elimination diets, removing inflammatory trigger proteins, which could be from the grains or from dairy, has almost magical, miraculous turnarounds, which surprise me too. Be it autoimmunity, be it digestive disorders, or outside the digestion, we look at conditions like respiratory conditions, mental health, depression, and diabetes, metabolic health, neurological conditions, literally everything. And when Dr. Alessio Fasano said, what are the 300 conditions? He said, they go under different names: diabetes, heart disease, metabolic disease, hypertension, autism, cancer, etc. And so, yes, this is something which is the future as far as I see. The use of probiotics is absolutely critical to repair the leaky gut, to restore the tight junctions. And today we also have laboratory testing for certain proteins which are so implicated with the leaky gut, which include zonulin, occludin, and antibodies against LPS and Candida. These are all also now going to be available in our country. So these markers are certainly there. And zonulin as a protein, and zonulin and occludin are triggered by gluten in wheat and some other grains, as well as bacterial dysbiosis. So yes, apart from that, we know that they have an immunomodulatory effect, and things like lactose intolerance we can treat through giving Lactobacillus bifidobacterium. And so the use, and like I think yesterday there were sessions, Dr. Pratisha also mentioned that early infancy and pregnancy, we can modulate the risk to allergies in children early in life. So the applications are only widening, and the use of specific strains is what I see as the future, meaning
as we go deeper into the understanding of the microbiome, as so many deliberations have happened in the last 24 hours on this, that we would be able to target customized and personalized solutions with specific strains. And I do see that as a future. And also, I was reading somewhere, desensitizing the allergen before it causes inflammation would be something potentially very, very useful and is expected to happen. So yes, it's extremely promising, and it seems to be the way things should be changing for us.
So I think about 2000 years before Alessio, Hippocrates said that all chronic disease begins with the gut. So
Yes.
in a sense, with our understanding, that might be true. So you advise the people who come to you for counseling, you advise them to avoid grapes. Would you advise them probiotics?
Yes, almost all my prescriptions when I see a problem is accompanied by a probiotic, be it a digestive disorder or be it leukoderma, vitiligo, they're all accompanied with probiotics. But we are at this point not in a position to give specific strains. So it's shot in the dark, whatever we think works for us and is available easily. Very rarely have I given a very specific strain because they're not even so easily available. And I'm going to take this opportunity, if you allow me to share. I mean, I've been sharing it with my colleagues, a case of an 18-year-old boy who came to me in August with an HbA1c of 15. And this boy was 108 kgs when he came to me. What I did was we gave him probiotics. We gave him an elimination protocol, which also meant change of grain from wheat to millet. And that's all I did. And this boy had been prescribed insulin by his treating doctor. So he did not want to go that way. So he preferred to follow the diet. And his inflammatory marker CRP was 7+. He had extremely low levels of vitamin B12 and was deficient in vitamin D as expected. Hemoglobin was excellent, 15. And within 3 months, he came back in November with an HbA1c of 6.3. And just now in February, he's just come back with an HbA1c of 6 without no drug, no insulin, and no change in weight. So he continues to be 108 kgs and he looks the same, but he has brought down. So what basically the point here is that do we treat diabetes as a metabolic disease or an immuno, is an allergen? What do we call it?
So that's very encouraging.
Yeah.
So I'm going to change direction a little bit. So I've had, two people in my family in my distant family who had chronic kidney disease on dialysis, and they are both prescribed probiotics called Loban. Basically, the idea being that it reduces blood urea nitrate. So I'm going to ask Dr. Shashikaran for a regulatory viewpoint on this, and I'll ask the clinicians what they feel about this.
Now, as far as food regulation is concerned, it will have no involvement as far as clinical management is concerned. Now, for example, if
this product is sold,
let's say CKD,
product, yes.
now if probiotics is advised for chronic kidney disease as an advantage, then it will go under a category of food for special medical purposes, okay, or food for special dietary uses. Under the food for special medical purposes, you need a physician's prescription to prescribe that food, which actually has probiotics in that. But if you want it to be of a generic nature, let us say people who have borderline kidney disease or something, and if it helps, they should be able to buy off the shelf. Then it comes in another category called foods for special dietary uses, where the label does not, the food does not require any prescription, but it says it is meant for a specific health condition. So there are that kind of category. The general category, of course, it cannot make any kind of a mention that it is meant for a disease. So the moment you say that food is meant for a disease or meant to treat or prevent a disease, it goes under the drug regulation. So there is a thin line between the probiotics going into the drug or coming into the food, and everything depends upon the intention for use.
Dr. Balla, you have any comments on that?
From a clinical standpoint, there are several situations in clinical practice, both adult and pediatric, where even if they are immunosuppressants, you can still go ahead and use probiotics. But I would be try to understand and use it with caution, because it can be sometimes a double-edged sword, especially if there's immunosuppression going on for some time. Choosing the right probiotic strain in the right situation would be important, probably.
I think probiotics, I have the other issue about what is available in the market and what is the actual content in it. So lots of times it's just a placebo. The claimed value is 6.5 billion spores. You check it, it's not even 65,000. So whatever he's receiving, it may or may not be working. But for CKD patients, I have a thing that in our country where they're using PPIs, which are available over the counter, why are we trying to regulate a probiotic, which is almost harmless? We'll have to focus on more on that part. And probiotics are not going to do any harm unless a person, like Sarat said, is immunocompromised and using spores which may cause a problem. But even in trials for IgA nephropathy, they have used probiotics in very high values, and they've had the safety profile was absolutely good. So there's no issue there. So specifically for your patient, the way you said, it's very, very safe. But for what it is being given, I don't know. It's not going to reverse the CKD. It may help in preventing in our tropical country diarrheas, or it may improve the immune system, but I really don't know how it will be effective. So specifically, IgA nephropathy, there are some places, there are trials, but nothing more than that, which I know.
Yeah, but the point is, per se, immunosuppression itself is not a contraindication to using probiotics. Several liver and kidney transplant recipients have been receiving probiotics also.
So the other thing that you're all faced with, we heard about a little bit about it today. Probably I should wind it up with this after this. Is that aging is probably one of the last frontiers eventually, right? So what do you think about how probiotics are going to be used to allow us to age gracefully? Any of you?
I think if you look at the mechanism of action of probiotics, they work everywhere. And if you link diseases, like she mentioned 300 diseases, I'll add another 50 more. There would be, it's whether it's a cause or whether it's a result, but the microbiome is not so great. And as you age, there is a decrease in level of, about bifidobacterium keeps going down. So there would be, scientifically, if you were to provide, there will be protection, there will be health, which will be better. I'm not talking about just gastrointestinal disorders. There will be cardiovascular health, mental health, neurological disorders. Everywhere it's going to be of great, great help. And using probiotics should be part of it. It should be, I feel, Yakult, I'm not going to name, should be part of a breakfast meal. It should be given elderly or anybody who is in that age group, it should be taken. So there is absolutely no harm and it should be used.
And may I just add that the current ecosystem with respect to our lives, and particularly in India, whether it's poisons in our food or whether it is modified food or it's the air, the pollution and the other toxic load of medication itself, I think the role of probiotics becomes even more critical.
Like the whole science evolved with Mechnikov's observation of a population which was eating a lot of fermented products and living up to a ripe old age.
And I also would connect with the theory of longevity of life, where the putrefying bacteria load was less, those who were consuming the fermented food. And through the probiotics, we can actually look the two aspects. One is keeping the gut health better, and immunomodulation, where the anti-inflammatory cytokines are produced more and pro-inflammatory are reduced. So we will not sick, or we will not be actually susceptible to any
lifestyle diseases.
All right, I think we've come to the end of this panel discussion. Just maybe if there's a comment or two from the audience, anybody has any questions to the panelists?
Probably also, Dr. Ramakrishnan, an institution like PGI, which is respected and recognized by the government and by the country, can take on such initiatives like fermented foods, which Dr. Prakash mentioned, and Dr. Tamang has been repeatedly talking about it, functional foods, fermented foods, clinical trials with that. Which is also locally relevant and relevant to the country. So it's much easier to initiate such a trial in an institution where there's a system in place. Thank you.
Yes, Dr. Bindu.
Regulation for probiotics based diet or supplements and stuff like that. They haven't done any approval for this kind of a product. I'm talking about FDA or EMA.
You say that there need not be any regulation for
They
probiotics?
say on their website, if you visit, they say that because there are a lot of questions being put whether probiotics, prebiotics, or a combination of that requires regulatory approval from FDA. They say no, FDA doesn't recommend any I mean, it's outside the domain. Maybe some way the way nutraceuticals are done with it.
Yeah, in fact, as far as our country is concerned, the probiotics and prebiotics both are under the nutraceutical regulation only. And the thing is, even when regulated, we have lot of fake products in the market.
Yes.
As Ajay Balla mentioned, the dose of bacteria present in a serve should be useful for the consumer. So if the dose is not...
FSSAI stamp, no, that product has FSSAI stamp. That's what I'm saying. FDA or any regulatory body doesn't recommend, but there are a lot of other loopholes which are present. So it's very good to say that this is good, this is bad, but what exactly is available in the market? Is that good? Is that regulated? It was just, an innocuous question that I had in mind.
Because the FDA's policy is a manufacturer of any supplement or a nutraceutical need not establish its efficacy. He only has to establish safety. So FDA is not even interested whether these supplements are working, do they work, etc., unless you make a claim. Whereas in India, the thing is there is a regulation, and in spite of regulation, we find there are a lot of lacunae, and we have a lot of products in the market claiming quite a bit and not having the sufficient amount of the particular probiotic which they are mentioning. So if it's unregulated, at least our country's environment, it will create quite a lot of problems.
It is.
Yeah. Just to add, in
Shashikar?
US, the FDA has got probiotics separately, and the rest are called LBP, or live biotherapeutic products. So LBPs are prescribed. They can be prescribed as clinical trials or as per the level of evidence based on that. So there are some products which doctors can prescribe, which will be given only, and these are LBPs grouped as that. Most probiotics are treated separately only as food supplements or food additives. So that's why the FDA regulation is slightly different.
Dr. Shashikaran, despite so many years of talking about probiotics, India, we still have this one question that is curd a probiotic? I'm taking curd or any traditional fermented food, then why do I need probiotics? That is one thing. And secondly, I don't know how many people are really aware about strain-specific health benefits of probiotics. So we see a lot of products in the Indian market. Of course, FSSAI has a regulatory framework, but nevertheless, we see probiotic ice cream, lactobacillus curd. They normally do not mention the strain of bacteria and sometimes not even the species of the bacteria. So would you like to just kind of throw light on the difference between your curd and fermented foods and probiotics, and also about the strain-specific health benefits of probiotics?
Of course, the curd is, what is marketed as curd, etc., unless they make a claim that it has probiotic benefit, then they need to follow the regulation saying that, how many bacteria, etc., are there. But you also find a whole lot of curds which don't make any such claims. So that is fine. But of course, it's a misconception that curd naturally contain probiotic bacteria. That of course only education or spread of knowledge will help. Now, as far as this strain specificity, the FSSAI has not given importance that whoever is manufacturing should mention the strain if it is claiming a particular effect, because most of the health benefits are going to be strain specific. Just mentioning the genus or phyla is not required. There's one more problem also is that you should not, it says not meant for children below 5 years, between 2 and 5. And when we know that the gut health is as important for infants and from the time they are born, and there is no reason why probiotics should be considered not to be recommended to children unless there's a medical prescription. That is one more error which has happened in that particular.
Actually, our conversation, we are always doing it in a very wrong way. What the regulators do, they set up a standard, and that standard is maintained. But essentially, any regulation will not change people's mind. The communication and advocacy is actually the tool which will make people make a choice and choose appropriately. So this is something all guys should do, that inform public properly, convince, try to make them believe what kind of science we have. If we have that kind of a language which they understand, not kind of a language we were speaking here, nobody will understand that. But so if we can make it understandable, then it will start working. And in Indian tradition, we should never badmouth curd. Even the Mechnikov and others didn't badmouth curd. Curd is a healthy food, so we need not say that it is a probiotic based food or whatever. It's a healthy food. So, and we have a lot of tradition. Don't take curd in the night. Don't take this and that. So we need to communicate properly. I have one question to Dr. Balla and one comment. Like what we were saying, Dr. Ramakrishna, that 25 years they changed. My list is different. Velcro, the Quartz watch, which does navigation, but one of the things which is with the probiotic, green bananas longevity, because this is now in all probiotic food. So whether it was Taiwanese, whether it is India, whether it is Chennai, wherever you go, this is one of the major, because green banana was always, the longevity of the green banana is what actually has made the difference. So this is very important for the probiotic based diet compositions. And Dr. Balla, whether you give colonic infusion or whether you give it through small intestine, if it is liver and liver-based diseases, it is given through small intestine. It is not given through colon.
No, I was talking about use in ulcerative colitis. Severe acute ulcerative
colitis, of course, colon.
Yeah.
With that, I think we'll. So there's just one question. One last question.
This was a very, very interesting session. I really enjoyed the session. I have one question to Dr. Prakash Halami. Are postbiotics better than probiotics or at par? Number one. Number two, have safety studies been done? Dr. Prakash?
Sir, yeah, quite interesting. Which I think two sides of the same coin. Probiotic has the advantage of its storage, as well as the transportation and the actual efficacy, where we can actually use only that active component and then take care. One so is probiotics, then viability is the issue. Then shelf life. So from that angle, I think postbiotics, once it is characterized, demonstrated the health benefit, one should go with that, with the postbiotic. Of course, now few examples which I have told here, which are commercially available, the safety issue has been worked out. Now the new molecule, once it is developed, its safety and efficacy has to be evaluated.
Actually, people are not aware about postbiotics. We have to create more awareness amongst the people.
Yeah, to some extent, just now related to postbiotic, we in fact have in the ILSIS a group, a working group about this postbiotic, where we had a discussion on this aspect. And now, traditionally, we will be consuming actually the postbiotic through the fermented food, which are actually cooked or pickled, but pickle actually bacteria will be live. But Professor Tamang knows most of this fermented food we actually cook. After fermentation, we cook or sun dry. So they will be actually will be the postbiotic product.
Yes, idli will have postbiotics because it is lysed well. Anyway, just continuing the discussion what Dr. Ganguly said, no, at FSSI, see, this is a body which you have to frame a standards and they will see that the standards are met. Now, what we have at present is simply guidelines that you can have these diseases. And if it comes that this genus and species is required, then the sample which will be taken by FSS food safety officer will check for that species only, nothing more than that. So please don't expect much in that. If you want to become more precise, it's a responsibility of the society and the stakeholders who are using it to give feedback to FSSI. See, the panel is 5-6 members who are sitting there. Probably they may not be having this. So it is better that we write to CEO FSSI that we want it like this. And these things are very seriously discussed because I am also there in panel since last 6 years. And we have lack of information. All the people sitting there, they're not really experts. So let those clinicians who prescribe and whatever difficulty they face should be brought to the notice of the CEO FSSI, maybe by email or maybe later. This is my appeal. Thank you.
Can I just suggest? Actually, the probiotic is an organism. It may be a bacterium, it may be a fungus, it may be yeast. So postbiotic is the effect, the products, the compounds. It is mostly the micromolecules, mostly the biotic compound, immunomodulators, vitamins, short fatty acid chain, and so many others, which have got so much health benefit. That is the difference. Now, the probiotics are also very important for the gut, the modulations. The final product is the metabolites. Mostly in the metabolites is the end product of metabolism. So there are two types of metabolites. One is secondary, one is primary, which is amino acid, fatty acid, and all. Then another is the secondary metabolites, untargeted. These are very important. These are the micromolecules. They can go to each compartment of any cell. So now the scientists are mostly concentrated on that. Now, regarding the probiotic regulation, Professor Ganguly sir and his team have already made a guidelines a long time by the ICMR, the DVT. But a lot of things have been changed and all. Now the FSSI has come with it. There are 25 scientific panels. In that, there is no probiotic panel. Though I'm a chairman of the alcoholic panel. Daruka chairman. Daruka chairman bana diya. So there in the thing, the regulation comes in the nutraceutical. So nutraceutical, we have already raised in the scientific committee. Why not you have the separate panel on the either Indian food, Amara Ayurved Ahar, Indian food or fermented food. And there you have the regulations, the clinical regulations, the probiotics and everything. Now we are demanding, now it is under thing. It has not been revised and all. Now what Dr. Sarath has said that, any bacterium which is claimed as a thing has a certain scientific the guidelines. It is called the food grade. The food grade organisms has to go for the several tests. One is the in vitro, as what Dr. Sasikaran, the Ramakrishna and Sasikaran has said that. Of course, the Professor Ganguly also said that In vitro, there are so many tests that includes the multi-omics also. But that is a in vitro, all the, the proteomics, transcriptomics, the metabolomics, lipidomics, and all these things and all. There are so many tests also. Then then very important test is the whole genome sequence. Within the whole genome sequence, we get an entire, you can annotate the entire map of the biomarkers and all. And there, one of the very important is the anti-AMR, the gene, the AMR gene, whether it is present or not, the virulence effect is present or not, that is passed. Then after that, very important, another is the in vivo animal models. And animal models, also, there are two types. One is the acute toxicity, which is just 7 to 10 days. And another is the subchronic of 3 months, where you have to do the both male and the female and test it. Then after that, only the human clinical trial, which we have not done, and we do not have any guidelines at all. In Japan and Korea, I visited, they have a, in Chungkokjang, in the Chumbuk National Hospital University, they have a very good the Center for the Functional Food, where they have a clinical trial. I was there as a 3 months as a visiting professor. They fit the fermented food of there, and they are 100-bedded room, and they do all the volunteer subjects.
I think we'll have to call
These are
this
not
off
there.
now. We are running.
No,
late.
my
We'll stop
suggestion,
now.
what is the, I think we can start this in the
Yeah.
PGIMR.
During lunch,
We
we
can
can
start
do
it.
that.
Thank you.
So thank you very much to the panelists for these discussions. I think we'll conclude this session.
And Neerja Hajela ji, thank you so much. I think it was a delight for a person like me to hear something which is good for health, rather than only hear what disease we should.
Thank you. Thank you.
Thank you. Now I request Professor B.S. Ramakrishna and Mr. Takeshi Yoshimoto to felicitate our panelists. As panelists, we have Dr. B. Sasikaran, once again, I request Professor B.S. Ramakrishnan and Mr. Takeshi Yoshimoto to felicitate our panelists. As panelists, we have Dr. B. Sasikaran, Dr. Prakash Halami. Let's have a huge round of applause for our panelists. Let's have a huge round of applause. Professor Ajay Bhalla. Dr. Sarath Gopalan. Let's have a huge round of applause. And Miss Ishi Khosla. Once again, let's have a huge round of applause for our panelists and our moderator. Thank you. Now let us come to the most awaited part of the day, the Young Investigator Award. On the basis of the oral presentation, we have our top 3 investigators with 1 consolation prize. For the same, could I please request the members of the jury, Dr. Osamu Chonan, Dr. Junji Fujimoto, Mr. Kosuke Kato, and Professor Patricia Conway to kindly join us on the stage for this special occasion. Let's have a huge round of applause for our jury. May I request Mr. Kato to announce the consolation prize and give away the award?
I'd like to say first one. The first one is Shuheta Gurelia.
Let's have a huge round of applause.
Congratulations.
Congratulations. Let's have a huge round of applause. Thank you. Now I would request Dr. Fujimoto to announce the 3rd prize and give away the award. Oh, acha, we were discussing consolation prize. First, I had to announce the consolation, then 3rd prize, then 2nd prize, and then the 1st prize. The one who have received the consolation prize, we announce that name first. Then we announce the one who have received the 3rd position, then 2nd, then 1st.
The consolation prize, Akashi Imara Kaharu Waru. Sorry.
That's good. Let's have a huge round of applause for the winner of the consolation prize. Now I request Mr. Kato to announce the 3rd prize and give away the award. 3rd prize.
The 3rd prize goes to Ms. Mitali Inamta.
Let's have a huge round of applause for the winner of the 3rd prize. Congratulations. Thank you. I would like to invite Professor Conway to announce the 2nd prize and give away the award.
So it's a pleasure to announce it's actually a tie for the 2nd position. And so I'd like Mr. Ahmad and Mr. Jaydeep to come.
Let's have a huge round of applause. Congratulations. Congratulations. I would now like to invite Dr. Chonan to announce the first prize and to give away the award.
Okay, so on the all presentation, young investor presentation is very, very nice. I'm very impressed and very difficult to choose the first one. So, and I'd like to announce the first one. It's the Shrita Gunglia from Chandigarh.
Yes, let's give her the huge round of applause. The winner of the first prize. Many, many congratulations. It's a huge round of applause for the winner of the Young Investigator Award, first position. Thank you. Now I request all the young investigators who presented their speech to come and join us here on the stage for a group photograph. As we have all our young investigators here who have won, can we have a huge round of applause for them for their excellent work? Thank you. Yes, let's have a group picture. Thank you. One special request to all the foundation members of Gut Microbiota and Probiotic Science. Please be present at the seminar hall first floor for the governing body meeting by 3 p.m. And now we all rise for the national anthem. As they say, all good things come to an end, and this two-day scientific feast comes to an end too. With this, we rise for the national anthem. Thank you so much, everyone, for joining at this wonderful scientific feast with us. The lunch is open. You all can enjoy your lunch. And once again, special request to the foundation members to please be there at the seminar hall, first floor by 3 p.m. for governing body meeting. Thank you.